अभी हमारे पास ये वाला केबिन वाला ना
Respected Chief Guest, Professor N.K. Mehra, Director of THSTI, Professor Pramod Garg, other dignitaries, faculty, scientists, and students present here. Good morning, and I would like to welcome you all at Translational Health Science and Technology Institute for the annual celebration of National Science Day. National Science Day is celebrated in our country on the 28th of February every year to commemorate the discovery of the Raman effect by Indian Nobel laureate Sir C.V. Raman. It is celebrated to encourage the scientific community to implement new ideas and technologies and to display efforts and achievements in the field of science for human welfare. The theme for National Science Day of the year 2022 is Integrated Approach in Science and Technology for Sustainable Future. This day marks the memory of a great scientist and provides us with the opportunity to honor his legacy. With this, I would now like to request you all to put your hands together as I request our Honorable Chief Guest, Professor N.K. Mehra, and Professor Pramod Garg, Director of THSTI, and Dr. Shinjini Bhatnagar to come forward to the stage for lighting the lamps. Please. Thank you, everyone. I would now like to request Dr. Nitya Vadva to kindly come forward to the dais for the welcome address.
So should I start? Good morning, everyone. On this important day, it is my honor and privilege to welcome our esteemed chief guest and keynote speaker, Professor Narendra Kumar Mehra, Honorary Emeritus Scientist of Indian Council of Medical Research, Vice President International Affairs of the Indian National Science Academy, and former Dean and National Chair of the All India Institute of Medical Sciences, New Delhi, Professor Pramod Garg, the Executive Director of Translational Health Science and Technology Institute, Professor Shinjini Bhatnagar, a Professor of Eminence at THSTI, esteemed members of the governing body, THSTI Society, Scientific Advisory Committee of THSTI, and the DBT officials who are present online, faculty and students of Hansraj College, Maitri College, Sri Venkateshwara College, and Manav Rachana International Institute of Research and Studies, my colleagues and students from both THSTI and RCB, those who are here in person and those who are attending online. As we celebrate the National Science Day, I would like to invite Professor Pramod Garg, the Executive Director, to welcome the dignitaries and address the August gathering. Welcome, sir.
Thank you, Nitya. Good morning, everybody. Respected Professor N.K. Mehra, chief guest for today's function. My senior faculty colleagues, scientists, Mr. Santo, our administrative and research staff, my research fellows, PhD students, all the students who have joined from 4 colleges, our esteemed members of society and governing body and scientific advisory committee who have joined online, and all others who are either here in person physically or online. I welcome you all to this important day of Science Day celebration at THSTI on this morning. It's my real pleasure to welcome Professor Mehra, who has kindly agreed to deliver today's keynote lecture. Professor Mehra, as you know, is an eminent scientist who has worked, I think, for last about 50 years in pursuit of science. He's someone who has contributed originally and has done fundamental research, particularly in the field of HLA. As you know, that transplantation has changed the lives of people who have been suffering from renal failure, and subsequently now transplantation is being done not only for renal failure, but now liver failure, heart failure, and others. And for that, you need an excellent cross-matching and making sure that the transplanted organ is taken up by the recipient's body. And in that regard, having your own capabilities and establishing an excellent facility at AIIMS was only done by Professor Mehra. And subsequently has trained a large number of scientists who have taken up this and spread it across the country. So he's widely recognized for his original science contribution. And I'm sure most of you know that in last 2 years of COVID pandemic, he's the one who has been teaching and educating even lay public through regular columns in Hindustan Times, which I have read all of them. And they are such beautifully written that anybody can understand even the intricacies of immune responses to virus infection. So, sir, thank you very much for those very nicely written lectures and articles in the newspaper. And otherwise, he has now come up with a book. And I'm so thankful to him that he personally presented me that book on immunology and particularly viral infections, sir. So as you just heard, that National Science Day is celebrated on this day in the honor and memory of Professor C.V. Raman, the first Nobel laureate from our country, for his original and fundamental discovery on Raman spectroscopy. A discovery which has actually transcended the boundaries of physics, chemistry, mathematics, and biology. And this is one discovery which is being used even today in so many diagnostics in medical field that we all know its importance after about 90 years of its discovery. Celebrating any particular day is so very important because it tells us about the important discoveries that have been made around the world and in this country, and would inspire our youngsters to take up that kind of science which will result in paradigm shift. So I think celebrating such events is very important. This is not just to have, it's not just a meet and greet session. It is something that will definitely inspire our youngsters. And you should remember those times when he made the discovery, there wasn't much in this country in terms of resources, money, or equipment. Today, you're so fortunate to have one of the best resource labs, not only in this institution, but across the country. And that is why you can make those discoveries and inventions which earlier people in this country couldn't do. One of the things that I always say is that one of the limitations that we have in this country is our inability to think big. And I would urge all of you to think big. Not just what others have done. That Me Too approach in science has no takers. You have to think and you have to challenge what is so-called accepted theory. Or although this is known. No, this is not known. You ought to make sure that this is actually known, this is actually true. You have to paint on a larger canvas and give original ideas and concepts. And I think it is not difficult to do. So let me give you some examples from this institution. So recently, sir, I'm very happy to tell you that THSTI got a very big grant from Coalition for Epidemic Preparedness Innovation. This is 12.5 million grant to develop a new pan-beta coronavirus vaccine. Now, this was a highly competitive grant, and I think we were 5th or 6th globally to have been awarded this grant. This is an entirely new concept. We are not following someone else. We also have now at least 7 or 8 molecules which are very promising against tuberculosis. The tuberculosis group here, which Ramandeep and Nishit and Amit and others are working on, and Dinesh, I'm sure one of these molecules will definitely go to the clinic, provided we provide them all the resources from taking this molecule from lab to the clinic. From the biology point of view of tuberculosis, Nishit has shown that beautifully CRISPR-Cas9 technology, which has now been used in so many laboratories around the world. This is something new thinking. So here also we can do. In the case of mother and child health, a large, probably the largest cohort in the world has been developed, and new findings are coming out of that. For example, new dating techniques, fetal ultrasound showing whether or not you will have a worse outcome of pregnancy or a good outcome of pregnancy, new findings in microbiome in pregnant women. See, this is something which is not Me Too approach. So here in this country, there are so many things that we can do. We have also taken new initiatives. For example, now earlier also, but now also we are a WHO collaborating center. We have now partnered with DNDi to develop therapeutics for dengue infection, which was, even today we have no effective treatment for dengue. So here, these are the examples which will tell you, you can do something what others can do in the West. In the case of immunology, for example, original work in Th9 cells have been done by Amit and his group. Original findings. So I think we can do definitely better than what we have been doing. And these events will encourage our youngsters to think big and also contribute to the growing science here. The theme for this year's Science Day celebration is integrated approach in science and technology for sustainable future. So let me briefly touch upon 2 keywords here. One is integrated approach, the other is sustainable future. So what do you mean by integrated approach? Now, integration is combining together the strengths of all the disciplines and leverage their individual strengths to come out with a holistic solution. To my mind, THSTI is an example of a model of this kind of an integrated approach. Here you have basic biology, you have mathematics, you have engineering, and the name actually suggests that. We have medical science here. And we have, if you look at all the other departments and disciplines that we have, we are actually taking this integrated approach to solving the problems of medical sciences, many diseases. And if you have collaboration with other major institutions, you actually do that integrated science. And I'm happy that we have collaboration nationally, internationally with so many other organizations. Sustainable is today what we have in this country or globally should be sustained over a long period of time. We have a commitment for our future generation to make sure that they have available all the resources and we don't use them all now. Everybody talks about energy and other things, and this is very important. So that is why renewable energy or reuse and reduce and other phrases have been given. These are very important. I don't think we'll be able to reduce consumption because of our growing demands and population. And I think large percentage of society is still underprivileged. They are going to use whatever we are using today. So I'm not sure in India, at least we can reduce, but surely we can have reuse and renewable, and making sure that the law of conservation of energy is taken care of. Energy doesn't go away. It remains in whatever form. And that is why science is so important to lead us to that renewable and sustainable future. So I would like to welcome all the delegates, especially the students from 4 colleges, Shri Venkateswara, Maitri, Manav Rashna, and Hansraj. And I'm sure you will have a very good experience here and learn something about science, which I'm sure many of you will take up as your future. I will once again welcome Professor Mehra and thank him for accepting our request to give the keynote lecture and be the chief guest. Thank you, sir.
Thank you, sir. I now have the pleasure to welcome Professor Mehra to deliver the keynote lecture. Professor Mehra is an honorary emeritus scientist of ICMR, the Vice President, International Affairs of the Indian National Science Academy, and former Dean and National Chair of the All India Institute of Medical Sciences, New Delhi. He joined AIIMS as faculty in 1979, rose to become Senior Professor and then Dean Research. He is an internationally acclaimed expert in the area of transplant immunology and clinical immunogenetics, for which his singular efforts culminated in establishing this specialty for the first time in India, thus facilitating solid organ and hematopoietic stem cell transplants in the country. His research focus has been on defining biomarkers of disease susceptibility in infectious and autoimmune diseases, influence of HLA matching and alloantibodies on transplant injury, stem cell biology, and regenerative medicine, among others. He played an important role in preparing the national guidelines for stem cell research in India. During the COVID-19 pandemic, he played a major role in public education and published a white paper, COVID-19 Host Immunity and Vaccines. Professor Mehra has been the president of the Indian Immunology Society, the counselor for the International Union of Immunological Societies, co-chair of the IUIS Gender Equality Committee, and founder secretary general of the Federation of Immunological Societies of Asia Oceania. He is a fellow of the Indian National Science Academy, National Academy of Sciences, National Academy of Medical Sciences, member honoris causa of the Hungarian Academy of Sciences, and fellow of the World Academy of Sciences. He served as chief editor of Frontiers in Immunology, special issue on clinical relevance of antibodies in solid organ transplantation. For excellence in medical research, Professor Mehra has received over 100 awards and academic honors. The French president conferred on him the title of Chevalier of the National Order of Merit. He also received the highest international award from the Iranian Research Organization for Science and Technology. He was a member of the international jury for the Else Kroner Fresenius International Award in Immunology and CIHR IRF Joint Research Projects. Professor Mehra and his group have published more than 490 original research papers in leading international journals. His book, The HLA System in Medicine and Biology, received high international acclaim. He has trained several PhD and MD students in the area of clinical immunogenetics and transplant immunology. I would request Professor Mehra to please come. Thank you.
Thank you very much, Nitya. Thank you so much for those very, very kind words. I'm so very happy to come to this institute. I've come here many, many times. And every time I come, I see that there is more and more growth. I was just telling Dr. Garg that every time I come and I see that the number of cars in the campus are growing, which indicates that the number of people working are increasing. So at the very outset, I want to thank Dr. Garg for giving me this opportunity to come here. I think celebrating National Science Day is a day when you can also look back to your own career as well. But I want to begin by giving homage to my friend who is actually no more, Dr. Maharaj Bhan. This institute was established due to his vision. And I was associated with the right from the beginning when the foundations of this institute was being laid down. And I'm so very happy to see a beautiful auditorium like this, beautiful buildings around, BSL 3s, SAF, and you name it. I think you are in the best of the place, not something like when I started my scientific career in the early 1970s. So today we are gathered here. Can you, how do I start my slide? I thought I will bring some slides. I'm very happy that in addition to the scientists of the Institute, Dr. Garg has also invited a lot of students. I'm told there are about 4 colleges in Delhi whose students are here. They're probably joined online. Yeah. And so I thought I will take some slides and tell you some stories of very prominent scientists in the past. And that might encourage you to see what you can do. The very first National Science Day was celebrated in India on February 28, 1987. This is to honor the invention made by Sir C.V. Raman. Sir C.V. Raman, as you know, was the most highly decorated, the most highly talented Indian physicist who was awarded Nobel Prize in 1930. That was the first Nobel Prize in Science that India had for his groundbreaking discovery on the light scattering phenomenon there. And we all know the Raman effect. I'm glad that in Bangalore, they have created a Raman Institute as well. The day provides hope and inspiration to future generations of scientists to pursue their goals. Several programs are being done all over the world, all over India. You know, when you are in science, you have children, you know, your students are like your children, and you have grandchildren, and you have great-grandchildren. And, you know, because the mantle has to pass on to the others. And therefore, I'm very appreciative of the fact that the institution thought about also having some students from colleges who are in class 11 or 12. And I think that's a very useful thing to do because many of them should get encouraged to take up science as a career. Of course, science and scientists came into prominence. You saw that during the COVID-19 pandemic. Everybody in India now knows the role that a scientist can play. And they thought, and probably today there is more of, the responsibility on your shoulders than any time in the past, because the public at large thinks that the scientists are the future of the country. It is due to the efforts of the scientists that we could overcome this pandemic. Remember March 2020, when the SARS-CoV-2 virus was first time found. We had only one lab in India, National Institute of Virology, who could actually detect the virus. We had no diagnostic labs. And today, with the efforts, indigenous efforts of the scientists in India, we have now more than nearly 5,000 labs, diagnostic labs. And they've all been authenticated through the Indian system, ICMR and all. We had no PPE kits. We had no ventilators over there. All, you can see that how India, through the use of science, could create a challenge into an opportunity. And this opportunity helped you. How many countries could make vaccine? India is only the 5th country in the world to make its own vaccine. You can think about a situation if we didn't have the vaccines, where were we? You know, we wouldn't have been able to vaccinate. Today, we have already vaccinated 180 million people in India. That's not a small number. I think this is remarkable achievement of Indian science. So therefore, what I'm saying is that the responsibility and the expectations of the public at large on you has become even more. And more youngsters who are in school or the early college state would be encouraged to take up science. So we commemorate this National Science Day to recognize the grandeur of science in everyday life, to eradicate taboos from common life, and to recognize the remarkable contributions of science in our day-to-day life. And today we have all realized how important science is in our day-to-day life. So the theme of this year's National Science Day, as the Minister for Science and Technology said the other day, integrated approach in science and technology for sustainable future. Integrated is the word. I was very happy when I came, I saw that one of the buildings you have named it as the integrated building. That's fantastic. So it is time now today to sustain that spirit of science that the common man has seen in this country during the pandemic and mark qualities of a good scientist. Now, when we talk about the qualities of a good scientist, I must take you to this guy, Sir Charles Darwin. I think if you read his story, he is probably the greatest scientist of all times. He discovered the theory of evolution. And how? Just through his keenness. These are the qualities of a good scientist. I have read this history of this guy, life history, a few times. And I marked these 4 or 5 things that he had. Very observant. And that's the quality that a good scientist will have to develop. Observant. You may not need very fanciful equipment, but you may have something that you are ignoring is already staring in your face, unless you're observant. Observant like people who have really achieved so much in life, made large collections of specimens. That was his quality. He would always collect specimens and make them. Today we are talking about a biobank. I think that there's a need for you to have a biobank. But what is more exciting is, and which really excited me, he always made extensive detailed notes. This is something that I see that in today's scientists, this quality is dwindling down because everything is on your screen, everything is on your mobile. People don't go to the library anymore because the library is in your hand. I remember when I started my science in the early 70s, we had a small library named Ames was a small institution at that time. And our library was on the ground floor. And a lot of people used to go to the library over there. We could see some of our very seniors regularly go to the library. We were also youngsters. You go to the library for many things, you know what I'm talking about. You meet friends over there. You meet your future partner also there. So going to the library is a very important aspect of any scientist. And you make notes. And this habit has stayed with me until today. If Dr. Garg is giving a lecture, I would sit over there, I'll start to make notes. Making notes, and look at this guy. I think whatever he could do, made extensive detailed notes. You can take a picture through your mobile or whatever you can take. But if you make your own notes with your own hands, this will stay with you for all times. He also was very curious and open to learning new things. This is the other quality that a good scientist must develop. But last but not least, he tried not to be biased and to base his scientific beliefs on evidence rather than on preconceived ideas. And we see this every day. Fortunately, now I sit on the other side of the table, giving the grant giving agencies and all that, and we sort of evaluate a lot of young scientists who come for a grant and all that. And many times you feel they come with a preconceived idea. It's okay to create a hypothesis. You must have a researchable question in front of you. And that question should drive you. What should be my objectives? What objectives should I make to fulfill this researchable question? and then what methodology I should use. It's not that somebody has done it, so I should also do it. So I think everything flows from one to the other. And these are the qualities of Sir Charles Darwin. Now, I want to take you to the turn of the century, and I've seen at least the last 2 centuries, the turn of the century has been a landmark year. And you see this 1901, 2 important events took place. And similarly, in 2001, 2 very important events took place. In 1901, when you entered the 21st century, the 2 important events were one by Karl Landsteiner, who discovered the blood groups. Now, imagine if you didn't have the blood groups, where was science actually going? And 1901 was also the year when all the work that Gregory Mendel did. Mendel, of course, started doing work in 1865. He was not a scientist, he was just a priest. And he was growing all these pea plants in his backyard garden over there, but he was very observant. The tall plant and the dwarf and this and the F1 hybrid and all. And he came up with some theories, which a lot of people didn't believe at that time. Until that turn of the century, there were 3 other scientists that said, oh, what Mendel was doing was absolutely correct. So all the work that Mendel did 35 years ago was recognized at that time. So imagine this turn of the 20th century was so remarkable year. Similarly, 2001, the turn of the 21st century, was a very remarkable year. A very high impact event took place. You had the Human Genome Project. You entered the 21st century with this book of genes in hand. And I made this slide, which I'm very fond of showing to everybody, particularly to my young colleagues. Look at this turn of the century, very remarkable year. Look at Greg Mendel and Karl Landsteiner. Both of them, by the way, are from Vienna, Austria. And I've been there a few times and went to those areas and went to see the lab where Karl Landsteiner were doing, and we discovered those blood groups over there. And then came the middle of the century, 1986. Of course, you know the DNA structure, the Watson and Crick, and many other. The last century was the century when the foundation of genomics was laid down. And today we are talking in terms of creating departments or centers of genomic medicine now. But one very important event took place, which has unfortunately gone unnoticed. It has not got as much importance as it should have, although it is affecting our everyday life. The PCR, the polymerase chain reaction, discovered by this guy, Kary Mullis, in 1986. And I want to tell a little bit of story about him. We met him, I and Dr. Ganguly, and I think Dr. Bhan was also there. We were in Berlin for some conference, and there he came and he gave a lecture there. So in the evening, we were encouraging him, gave him a couple of drinks. We wanted to hear his story. How did he develop, how did he discover this polymerase chain reaction? And of course, after 2 hours of discussion with us, we sort of felt he's a crazy guy. The scientist has to be crazy as well. His craziness was, he's a biochemist. Unfortunately, he died, Kary Mullis. He said, I as a biochemist could make the DNA 1 copy, a similar copy one day. I was very excited. I thought if I can make another copy, 2 copies, why can't I make 4? If I make 4, why can't I make 8? And we went on, and now he could make thousands of so. But what he told was that he used to be working in the lab like a crazy guy all the time, in his mind, the nucleotides and the DNA over there. He said he used to go home very late in the evening, and many times he would, because a bright idea comes to his mind while he's going home, he would just stop his car by the side of a river front, sit down over there. And we didn't have the mobile phones at that time and all. His wife would be waiting at home. And he said many times when I come home, I got a bucket full of bangings from my wife. I said, no problem. This is something that we do. Whether you get the Nobel Prize or not, wife to danti hai bhai. So there's no problem about that. But he said, because I would just sit over there, I won't even know how much time it has taken place. So you have to be crazy like him. But look at what he has done. 86, he discovered for the first time the polymerase chain reaction. And all that human genome work that you did in 90s, the 90s decade, we still call that decade has been the most remarkable decade of science. You know, you were spending billions of dollars in trying to understand the human genome, structural genomics and the functional genomics. And a lot of people were questioning at that time, oh, you're spending billions of dollars, what for? But then when you entered the 21st century, you entered with this book of genes in hand. And thanks to these two guys, Francis Collins and Craig Venter. Francis, of course, has been the director of the National Institute of Health in USA for several years. And when this book of genes came to hand, everybody started to say, we are now there to do big science. Journals like Science, Nature, had their on the cover page, scientists are now there to do big science. So whereas the 20th century was the century of foundation of genomics, I think the 21st century is the century for doing big science. We have only gone through 2 decades yet, but there is so much more to do. Because the tools and technologies that you have in hand, nobody had last century. So I think you're born in a very lucky era. The third story I want to tell you about these two guys, originators. We call them originators. These were originators of what we call major histocompatibility complex, MHC. And those of us who are immunologists know the importance of MHC. And so much so that people who got the Nobel Prize, they said, we are here because MSC was there. If MSC was not there, or in the human system, the HLA system was not there, we were not there. And these guys discovered the concept for the first time in the mouse. What they were doing in the early 30s, Peter Gorer from UK, Lister Institute in London, a young guy, they were transplanting skin, they were grafting skin grafts from one strain of mouse to the other, trying to understand, is this something that controls the success of the graft? And similar work was being done by George Snell. George was in Maine, in USA, the Jackson Laboratories, the famous Jackson Laboratories over there, a senior guy. And then, of course, they met through, I don't think there were phone calls at that time, somehow they met. And Peter Gorer then went to US to work in his lab. And they defined that there are at least 20 systems, or 20 systems of what you call transplantation antigens, as they called, which control the success of a graft. If you're grafting from one strain of mouse to the other, mind it, these are inbred strains, which is something that you can't have in man, but strain A to strain B and B to A, and they were trying to understand what controls the success of this graft. And George Snell, of course, was a senior person, but all the work was done by Gorer. And Gorer was the one which came up with the terminology histocompatibility. They were doing tissue transfer. Skin is a tissue. Compatibility. So tissue in scientific terms called histocompatibility. And he said, out of this 22nd is the most important one. He called it H2 system. And these are the originators. And by 1948, this terminology was born. There's something called major histocompatibility complex, which at least in the mouse is H2. Nothing was known in man until these guys came into the field. 58. A French scientist, look at his story, remarkable story. Jean Dosset was a physician. He used to go to the ward rounds and he would look for people who required more than one blood transfusion there. He started collecting sera from those people and trying and see if they would develop antibodies, because they are getting blood transfusion from a third-party source, many. And lo and behold, he said, yes, we found that. But this guy from Holland, Jan van Rood, Jan was a Dutch scientist, and actually my whole training is done by Jan. I must have, at some point of time in the 70s and 80s, Holland had become my second home. I was probably traveling over there 2-3 times a year. And Yon argued if the multi-transfused people can raise antibodies, then the multiparous woman should also do. Because she is harboring the baby for 9 months in the womb, half the genes are from the father, so she should raise antibodies. So all this led to the birth of this system, the birth of HLA, a system of prime importance for organ transplantation, even till date. It is not only important for organ transplantation, kidney, liver, heart, also for the hematopoietic stem cell transplantation. So, you know, how these people, and if you read their papers now of 1958, आप सोचेंगे क्या यार बड़ा simple सा experiment तो किया था. But you have to go back to that year and see those years, they had nothing else. And I thought towards the end, because when I talk of Yon Van Rode, Yon was a remarkable guy. And I thought I should also tell you a little bit about my story. About 5 decades ago, as I said, in the early 70s, we just used to go to the libraries and read books. We had no other way of keeping ourselves updated with science. And some of this lymphocyte excited me. But when I went to the library, there was a book in the library written by this very great guy, John Yoffe. Yoffe was a great scientist from the Oxford University. And he wrote a book, The Lymphocyte. It was about 200 pages or 270 pages, something like this. And I read from one page to the other. And that was my first real rendezvous with the lymphocyte. But somewhere Yoffe wrote, small lymphocyte must be an end cell. And why did he say so? Because that time there was no electron microscope. There was no transmission electron microscope. There was no scanning electron microscope. He used to see the lymphocyte under the light microscope or oil immersion lens. And small lymphocyte, when you see under the light microscope, is just a dark cell when you stain it with hematoxylin and eosin, with a very small rim of cytoplasm there. No cytoplasmic organelle. And any cell biologist will say, if a cell has no cytoplasmic organelle, iska to kuch kaam hi nahi. It's an end cell. And today, immunologists, I don't know whether Amit is here or not, he talks about at least 20 subtypes of lymphocytes. And during the COVID-19, and my school students must pay attention to this, that everybody on the street was talking about the role of lymphocytes. T and B cells, the antibodies forming cell. I got the vaccination, or I got infected. I should have raised antibodies and all. So look at the concept changing from, and then of course the electron microscopy came, and you could see under the electron, but even under the electron microscope, the small lymphocyte is a very small rim of cytoplasm, maybe 1 or 2 mitochondria there, and you will not call that cell as an active cell. But it is the most important cell that we have in the body there. And I was, in the early 70s, we were trying to induce, leprosy was very common in India at that time. You know, today's generation probably doesn't know that in the early 70s, 74, 75, WHO came up with the data that about 10 to 12 million cases of leprosy all over the world. 5 to 7 million were in India. And every symposium, the workshop that will take place in institute in AIIMS would be directed only towards the infectious diseases. And infectious diseases, it has to be leprosy there. But leprosy was a remarkable disease. If anybody wants to take immunology as a career, you must learn leprosy. On one end of the spectrum, you got the lepromatous form, and the other you got the tuberculoid, and then you have the fluid spectrum in between. Very, very important. And you see the dichotomy of the immune response over there. So we were trying to induce leprosy in the mouse model. I got those 4 pairs of the CBA-J strain from Jackson Laboratories, USA. We didn't have money more than this. 4 pairs, we were trying to breed these now, want to make more in the colonies. That's how we sort of developed the expertise. We could have colonies of these mice, but then doing thymectomy in the mouse, total body radiation, bone marrow reconstitution, and then give the mLepre inoculum. Well, it's difficult work at that time. And even thymectomy itself was not. And I must have done, thank God there were no ethics at that time. I must have done about 1,000 thymectomies in the mouse neonatal as well in the 10 to 12 weeks of mouse. And, this took us to the leprosy field. This took me to this area in Wardha, Sevagram, central part of India. You remember Gandhiji spent about 10 to 12 years over there. I'm talking of 74, 75. Leprosy was prevalent there. You go from village to village, every household will have at least one case. Large family size, maybe 2 children would have a leprosy, not all. So that raised the question, is there any role of genetics here? All of these people are living in the same environment, they're sharing clothes, they're sharing food, yet only 2 children develop leprosy, others don't. And 1973 was a remarkable year when Bruton in UK for the first time showed that this HLA system is not just only for transplantation, it's also a very good biomarker. Because he showed patients with ankylosing spondylitis, almost all of them have this gene called HLA-B27. So this gave a new dimension to the whole field. And by this time, Rolf Zinkernagel and Doherty, they showed for the first time there are immune response genes. So that gave a whole new dimension to the whole field. So somebody said, why don't we study HLA antigen distribution in patients with leprosy and this? Very difficult task. I didn't have any equipment, I should tell you. I had only this. I had a simple monocular microscope. While for this kind of a study, you need an inverted phase contrast microscope. I didn't have neither the money. Didn't have the micro syringe. I think I must have stolen from somewhere. And the plates, the micro test sprays. These are very common things for you in the lab now. But the sera was not there, the serology-based test. And I could learn from the journals that you write to the NIH Serum Bank. I wrote to the NIH Serum Bank, no, those days, chithi jati hai, so it takes about 15 days for it to reach there, and maybe 1 month or about 2 months after the letter came, yes, we will be very happy to give you sera. I then wrote, please also send me some trays. And they sent that. And also a letter came stating, we are very happy to know that somebody from India wants to do a study on this. I had no training. I just looked from the journal, and you can see I didn't have a microscope, but I did this study. The patients from Wardha area or from Safdarjung Hospital and AIIMS Hospital. And my first paper got published in 1975 in this top HLA journal called Tissue Antigens. Mind in volume 5, number 5. I mean, the journal had just come up. This field was just beginning to grow. 1973 was the year when somebody showed that there's an association of HLA-B27 with this. So I was almost at par with the world, but I was shaky because nobody was training me. But there was nobody in India to train me. Then came this very important landmark in my life that Jan Dosse, whom you saw that he, through the blood transfusion and all that, 4 years before he got the Nobel Prize, he organized this first international symposium on HLA in health and disease, not for transplantation. This field was developed only for organ transplantation, but now it was moving towards the disease part. And I was very keen to go there to this symposium. We had no money. Fortunately, our director of the institute, Dr. Ramalingaswamy, was a brilliant scientist. And he used to mark his youngsters. Fortunately, I was in his radar. I went to him and said, I want to go to this symposium. I have done this work in leprosy, which is published as well. It will give me an opportunity to meet these people. He said, yes, the institute doesn't have the money, but I will talk to the British Council. The influence that he had, just one phone call, and I got the travel money. I went over there, my poster, HLA in leprosy, and then came this guy who has this, the smoking thing in his hand, René de Vries. He was also a youngster at that time. They had done some study on leprosy from Suriname. Suriname, as you know, is a Dutch colony. And they had done much more elaborate study than I had done. And I had gone to read their poster. I couldn't even understand. So like a enthusiastic guy, he came to me and said, "Oh, have you done this study in India?" I said, "Yes." He said, "Jan van Rood would like to come and meet you tomorrow. So be here at 1 o'clock. I can tell you I didn't sleep that night because Jan was being seen over there as a god, you know, the greatest scientist over there. But he came. He was very good. When he came to talk to me, you know, something, because Rama had told me when I was leaving, he said, he used to call me, Naren, when you are going, make sure that you keep the flag of India flying high. Now, this sentence has stayed with me all my life, and I want to pass it on to you people. Wherever you go, whatever you do, you are an Indian, keep the flag of India flying high. So when he came to me, I thought I should hide certain things from him. I think something in me then counseled me, no, he's too big a man, you tell him the truth. He came and said, like they normally do, "Oh, beautiful study, very nice study. Did you do this in India?" I say, "Yes." Very good. Then I was there to look for a place where I could do a postdoc. The people like this, or primarily I wanted to go to the US. He said, "We have done a similar study, a similar leprosy study from Suriname. Have you seen it?" I said, "Yes, sir, I have seen it. I couldn't understand it." So he said, and I said, "I don't know what I have done is correct or not. Nobody has trained me. I don't even know that the statistics I have used are correct or not." Well, we had no training of statistics at that time. And I told him correctly, "I don't have the inverted phase contrast marker. I just did it like this." He said, "How did you do it?" I said, "Because I didn't have the inverted phase contrast marker. I have to only see from the top." So I was doing this test and flicking the tray, you know, of the dye, and all the dye would come on my coat and all that. And this is how I have done. So I'm not sure what data I'm showing is correct, but it is published. He said, "Would you like to collaborate with us?" Now, this was a bolt from the blue, you know, collaborate with you? What do you mean? And then he said, "When are you going back to India?" I said, "The day after tomorrow." He said, "No, you're not going to go back. Come with us to Leiden, Holland. Leiden is the oldest university in Holland." He went away. And then this guy, Rene, came to me. I said, "Look, John told me you don't go back to India. Come with us over there. I have bought a very cheap air ticket. Can't be changed. I have to go there. Otherwise, I can't even go back to India." He said, No. Once Jan has said, you better come. And Jan took me in his car to Leiden. It was a 3-hour journey. And that is what changed my life. That is what I got drawn into the field of transplant immunology. What I saw when I went over there was eye-opener. The kind of labs they had. I had a small little room where I was doing small little thing. But that's the kind of a field that I have to go into. But, you know, this was remarkable. And then, of course, I came back and we wrote this grant for the WHO training course in HLA. We were ourselves needing a training. We were doing this training course. All that our intention was to get some money and get a microscope. And now I had this in inverted phase contrast microscope. It still stays in my lab in AIIMS, and my successors, you know, those children, grandchildren, and great-grandchildren are still very fond of this one. But my biggest day came when Dosse came to us in 1993. And that used to be my chair, and Dosse enters my room and he says, "You get up, you sit over there, I want to sit on your chair." This is the kind of encouragement these people give you. I couldn't believe it. He said, "You sit here, I want to sit there." And by chance, Dr. Dash, he was our head of the department of nephrology, and we used to meet very often, you know. Because of this field, I had interaction with almost every department in the institute, you know, surgery and gastroenterology, nephrology and oncology, everybody. And look at the way, and then we had a lecture from him. He interacted with my team. Many of these people are now settled in the US and doing very, very well. Colonel Chopra, GS Chopra, he had come to me as a lieutenant colonel. He then rose to the rank of Lieutenant General. Unfortunately, he died 2-3 years ago. But this is my story. And then, of course, from there on, there was nothing, no going back, no looking back, as you say. I was traveling all over the world, from one place to the other, and then training people, as Dr. Garg said. I must have trained more than 100 people. I'm very proud to say that today, even in the private labs, if there are any people who are doing HLA typing, they have rubbed shoulders with me, either as a student or a trainee there. We created, like in the US, they have the American Society of Histocompatibility and Immunogenetics. I said, we will create the Indian Society of Histocompatibility and Immunogenetics. And we do our conferences every year. We do our webinars and all that. That encourages our young. After all, your job is to now make sure that you have pool of young scientists who can take over this mantle, actually. And that's the most important part. So towards the end, I want to talk a few things to my scientific colleagues over here, the young scientists, the students. Always try novel approaches. Never be afraid of a novel approach. You should have conviction in your mind. What you need to do is to sit together. Gone are the days when we used to work singly, single person. I don't want to have interaction with anybody else. I think that's a wrong approach. And today you have so much of equipment all around, so much of facilities all around. You need to have interaction. And this is something that I saw for the first time in Leiden, Holland, when I went to over there. How these people on every Monday morning, 2-3 hours, everybody will come and interaction. If you have done any study, you better come and tell us the raw data and all that, how you will sort of interpret it. So develop a conviction for trying novel approaches. Don't be scared of failures. Failures will come. I think failure is a part of life, but you should have self-belief. You look at the history of all these big scientists, Darwin, Mendel, everybody has gone through failures. If the failures were not there, you're not going to learn. And the integrated approach. This is the theme of the National Science Day of 2022. The other day, Dr. Jitendra Singh was talking about that the government of India has already planned. There are 4 steps that they have said. I don't have a slide for that they want to bring all science organizations, not just life sciences, mathematics, the engineering sciences, all of them. They're going to integrate them into one. I think that's a great approach. This is something that we used to have. You remember, Dr. Garg, when I was the dean and you were with me as a colleague, we went, I took a delegation of 5 people to Holland, because Holland was very keen to develop scientific collaboration with AIIMS. Each of those universities that we went, we were struck by a beautiful new building around. हम कहते थे भाई ये क्या है building? So this is for the private enterprise. तो private enterprise के लिए separate building बना दिया. So this scientist, if you find something good and if you feel that it is something good, technology or I think during our time, we had many like this, but we had no way to go. We had no idea how to approach the private enterprise. By the way, at that time, it was a taboo. You are an academic institute, you remain in the academic environment. Don't talk to any private institute. And today we are talking public-private interaction is most important for taking this. A few things, and I want to show you this quote by Albert Einstein. He said, if someone feels that they had slide is this guy, David Baltimore. Hey, extremely important scientist. He came to AIIMS he got a Nobel Prize in 1975, by the way. He came to AIIMS in November 7, 2007. We had a lecture from him. And this is his quote, which through my habit of making notes, I sort of wrote this. He said, doing science at the highest possible level is one of the greatest forms of fun as there can be in this world. Read each word of this line. You get paid for it. People appreciate you for it. You contribute to society. And in my view, they just cannot beat it. I will be 70 years old next year, and I still have a new result as I ever did. It is the greatest high I know. So if you are in science, you've chosen a difficult path. Science is not going to sort of take you immediately to make you rich, actually, but it can enrich your knowledge a lot. You need to develop all of these qualities that I thought I should tell you through all these slides by looking at the history or the experiences of some of these scientists. Thank you very much once again for inviting me and giving me this opportunity.
Thank you, sir, for your enlightened speech and wonderful words. I would now like to request Professor Garg and Dr. Shinjini to step forward for the felicitation of our honored chief guest, Dr. N.K. Mehra. Thank you, ma'am, and thank you, sir. I would now request Professor Garg and Professor Mehra to please move forward to the first row. Moving forward, now it is time for the oral research presentations by THSTI PhD scholars. For this, Professor Mehra will be the judge of the event. And I would request all the participants to kindly be ready. Our first participant is Mr. Akshay Binayak. Please put your hands together, everyone.
Just a minute. Yeah. So good morning, everyone. I'm Akshay Binayak, and I am a PhD student working under the supervision of Dr. Amit Awasthi. And since the last 2 years, I have been working towards the COVID-19 acute infection studies. And the title of my presentation is Immunometabolic Signatures that drive and shape the antigen-specific T-cell immunity during COVID-19 infection. So we have entered the third year of the COVID-19 pandemic, and almost 5.8 million people have succumbed to this disease. And the pandemic is still not over, and numerous variants of concerns are emerging. And it appears that as the mutations occur because of the RNA nature of this SARS-CoV-2 virus, the mutations are largely in the, for example, in the Delta variant, there are 9 mutations, whereas in Omicron, there are 34 mutations, which eventually lead to a reduction in the neutralizing antibody responses. And therefore, the overall efficacy of the vaccines go down. but it is the T cell responses which are largely preserved among these variants. And therefore, we wanted to focus on the T cell responses. Importantly, for the controlling of COVID-19 severity. For example, in the case of mild diseases, it has been observed that the functional T cells remain largely are in larger frequencies in those who have mild diseases. Whereas when there's a person who has got severe disease, it tends to be that they have fewer functional T cells, whereas there is no such correlation observed in the antibody responses. Therefore, the T cell responses is critical in controlling the COVID-19 severity. So we wanted to focus, and from a conventional viral immunology point of view, a mild infection occurs when there is a strong and timely innate immune response, which gives rise to a robust T cell. So we wanted to study them for the antigen-specific T cell response studies and the innate immune response studies. And the plasma was used for humoral response and the metabolomic studies. And we tried to correlate the T cell immune response when it is largely the T cell response occurs. And the initial early time point of innate immune responses were then correlated with the metabolomics. To study the T cell response, we took the which recognize these peptides will then start secreting cytokines, and these cytokines and the activation markers were then evaluated by flow cytometry and the cytokine bead assays. So we found that as the time progressed, the T cell immune responses also increased. And more importantly, we observed that the T cell immune responses were largely conserved between the different variants of concerns, such as the wild type, Delta, and Omicron. And next, TLR agonists, such as the R848 and poly-IC, which are kind of specific to the SARS-CoV-2 innate immune responses. And then the culture supernatant was collected after 24 hours, and the cytokine analysis was performed. So we observed that when we try to correlate the antigen-specific T cell responses during the V3 with the innate immune responses, we observed that the cytokines such as that of IL-6, IL-23, and IL-1 beta, which are the pro-inflammatory cytokines, were strongly correlating with those of the antigen-specific T cell immune responses, therefore suggesting that a strong pro-inflammatory innate immune response were responsible or were strongly correlating with the robust T cell immune responses. In patients, analysis that the metabolomic signature of the COVID-19 infected individuals was largely different from that of those of healthy individuals. This can also be seen in this heat map, in which the itaconate, the azelaic acid, norvalin, pyroglutamate, and many more were which were involved in the histidine metabolism, arginine biosynthesis, D-glutamine and glutamate metabolism, etc. were correlating significantly with the T cell immune responses in the V3 or 14 days after the RT-PCR report. Therefore, to conclude, we found that an antigen-specific T cell response is elicited or is being strongly correlated with a strong pro-inflammatory innate immune response driven by IL-6, IL-1 beta, and IL-23. And there was a significant correlation between the itaconate, norvalin, azelaic acid, and pyroglutamate. These are the metabolites which eventually gave to a strong T cell antigen-specific T cell response. So, from a translational aspect, we wanted to study, see how can we apply this study, and one approach can be that this can eventually lead to a novel vaccine platform in which the use of TLR agonist, which will eventually give rise to a strong pro-inflammatory IL-6, IL-1 beta, and IL-23 based response. And in the presence of supplementary metabolites, such as those of arginine biosynthesis and the glutamine metabolism, will eventually give to a better protection against COVID-19 variants by eliciting a strong antigen specific T cell response. So these are the acknowledgements. I would like to thank my supervisor, Dr. Amit Awasthi, all the collaborators, and my lab and THSTI. Thank you. If there are any questions, I'd like to answer them. Mine? Yeah.
Very nice. Thank you very much.
Thank you, sir.
मुझे ये बताओ कि आपने सैंपल्स लिए V1, V2 and V3.
Yes, sir.
Your V1 was -3 days.
No, sir. 0 to 3 days.
हाँ तो वो 0 to 3 days after
After the
infection.
RT-PCR report.
After
Positive
infection.
RT-PCR report.
मुझे ऐसे लगा जैसे आपने स्लाइड में -3 डे लगा, तो मैंने कहा आपको कैसे पता चला कि इस आदमी को इन्फेक्शन हो जाएगा?
नहीं, sir, that's-
So what you are saying is goes down and is taken over by the adaptive immunity, right? This is what you're showing us.
Yes.
Adaptive immunity. However, in COVID-19, there was a specific, very strange phenomenon. The innate immunity doesn't go down in some people. It remains sustained. So therefore, you got a poor. If the innate immunity remains sustained, adaptive will not come into the
Right.
picture over there. Is there something that you saw in patients who developed severe form of infection?
Sir, the cohort which we
Have you seen that
followed-
paper by, what is her name, Mandana? I think came up in June 2020 or August 2020. Remarkable paper during COVID-19.
The cohort which we followed was largely of the mild and asymptomatic patients. It did not involve severe because we were trying to focus on what are the strong correlates of those who actually recover. So we try to focus on that. But interestingly, which a very good point which you have raised is regarding those who have got a sustained innate immune responses and do not go down. So the metabolites which we found, for example, the norvalin and the itaconate, These are the metabolites which actually bring down the levels of IL-6 and IL-1 beta experimentally proven by other studies. So this suggesting that these metabolites will
But I
actually.
wanted to know your V1, 2, 3. That's a very good way of looking at it. Were you, anytime you thought there is something called lymphocyte exhaustion here? या तो सेल थक गए हैं और या फिर वो कम हो गए. Even if you took only the milder form of infection, what was your observation in V2, V3 in terms of lymphocyte exhaustion or lymphopenia?
So, sir, we did not look into the exhaustion, for example, the expression of PD-1 or the other exhaustion markers which are expressed by the T cells. But we looked into the functional response, because if there is an exhaustion, it tends to be the expression of the cytokines such as the TNF alpha and IFN gamma are relatively low because of the exhaustion state. That we did not find, but absolutely this is an important angle to look into that we have not further studied.
From your study to the man on the street, everybody asks us, sir, मेरी antibody नीचे चले गई, अब मेरे को I don't know whether I'm okay or not. So with your T cell experiments, what will you guide in translational mode to the man on the street?
So the most important from the current scenario is that from this slide, if you see, all the neutralizing antibodies, I have not added the neutralizing antibody data over here, but the neutralizing antibodies significantly go down against the variants of concerns. But when, if you look into the Delta and the Omicron T cell response, and even in the vaccinated individuals, we have studied this in the other scenarios, in other studies, that the antigen-specific T cell response persists against those variants. So you don't have to worry much. You just take your booster doses when the time comes and believe in your T cell responses. And
ठीक है, my last question, मुझे ये बताओ, why do neutralizing antibody titer go down? नीचे क्यों जाता? आपने जब वैक्सीन ले ली, आपको इन्फेक्शन हो गया, एंटीबॉडी बन गई, तो आफ्टर सम टाइम 5-6 महीने के बाद नीचे क्यों जाती?
So there is a half-life of antibodies. So eventually the titer will go down. Although memory B cells might persist, but the antibody responses, they can again come back. The memory B cells might take care of that, but because there is a half-life of the antibodies.
भैया memory तो B cells में भी है. What cells make antibodies?
Plasma, plasma cells. Yeah.
Plasma cells, yeah. B cells turn into plasma cells and they make antibodies.
Yes.
Plasma cell की life कितनी होती?
Sir, I don't know. Sorry.
Plasma cell has a life 4 to 5 months.
Okay.
वो उनका they will go into senescence phase. वो तो नीचे जाएंगे तो एंटीबॉडी तो नीचे जाएगी. आप ये कह रहे हैं लोगों को, भाई don't worry, T cell है ना, that will take care of it.
Yes.
Okay, thank you.
Thank you, sir.
Anybody else wants to?
Any more questions, please?
Thank you.
A small reminder for the participants, the presentation time limit is 8 minutes. The first reminder bell will be rung at 7 minutes and then at the 8-minute limit. And 2 minutes will be given after that for Q&A with the judges and the audience. Next, I would like to call Jaskaran Kaur for the presentation. Thank you.
Good afternoon to one and all. Today I'm here to give a presentation on my PhD work thesis, which is entitled to study the association of dengue virus infection and megakaryopoiesis in an in vitro model system. Now, dengue virus infection can cause a self-limiting febrile illness, which can potentially turn fatal. And irrespective of the disease severity, dengue infection leads to thrombocytopenia to a lesser or greater extent, with severe patients exhibiting acute drop in platelet count along with vascular leakage. Now, multiple mechanisms working in parallel have been proposed to contribute to this thrombocytopenia, which includes attenuated platelet biogenesis or increase in the platelet decay. Which brings us to our research question, which is to have a mechanistic understanding of the association between megakaryopoiesis and dengue virus replication. Now, for this study, we had three objectives. The first one was to characterize a model system which could mimic the in vivo megakaryopoiesis. The second objective was to characterize the replication of kinetics of this virus in our model system and to observe how megakaryopoiesis influences the viral replication, while the third objective was to observe the effect of dengue virus replication on megakaryopoiesis. Now, talking about megakaryopoiesis, it is a process of commitment and differentiation of the progenitor cells into megakaryocytes, which is accompanied by profound changes in the molecular and cellular characteristics of the cells, which include increase in the cell size, development of polyploidy, accumulation of a lot of proteins and membranes, and expression of certain genes and surface markers. So the model system that we have used for our study is based on K562 cell line, which when treated with sodium butyrate differentiates towards erythrocytes, but with PMA treatment, it will start differentiating towards megakaryocytes. Now, for the characterization of model system, the K562 cells were either treated with DMSO, which served as the vehicle control, or it was treated with PMA, and the cells were harvested at day 3 and day 6. And the different characteristic features of the megakaryopoiesis process were quantified. And some of the results I'll be showing here. So as we can see, there was development of multilobed nucleus and polyploid cells with PMA treatment at both day 3 and day 6. And also there was accumulation of intracellular ROS and expression of certain megakaryocyte specific surface markers like CD4161 heterodimer and CD61 markers, which indicated that K562 cells recapitulate major events in megakaryopoiesis upon PMA treatment. Now, next, we wanted to see the effect of differentiation of these cells on dengue virus replication, for which prior to the PMA treatment, the cells were given infection with the virus, and the different crucial events of the virus life cycle were studied. So the accumulation of the intracellular virus antigen was quantified using FACS and 4G2 staining, and we found that the percentage dengue antigen positive cells were significantly higher in PMA treated cells as compared to DMSO treated undifferentiating cells at both day 3 and day 6. Now, to correlate the higher protein accumulation in the cells to the viral replication and virus release, we quantified the dengue virus RNA level by qPCR and the release of the virus in the culture supernatant by FFU assay, and found that in both the conditions, there was an upsurge in the viral RNA and the virus titer at both day 3 and day 6 after PMA treatment in the cells. Now, to rule out the possibility that the higher viral replication in the cells was because of the enhanced entry of the virus in cells which were actually differentiating, we did an entry assay, but found that the entry was comparable in both these conditions. And all these data together suggested that differentiation of K562 megakaryocytes promotes replication of dengue virus. Now, megakaryopoiesis is associated with a dramatic change in the gene expression patterns in the cells. And to further investigate this, so this might be creating a better milieu for the virus to replicate better in the differentiating cells. So to further investigate this, we did the transcriptome analysis of the differentiating cells with and without infection. And from the data, we found that there was enrichment of a very crucial transcription factor, NFE2L2, which is also known as NRF2. It is a well-studied transcription factor, which plays an important role in the cells in maintaining the redox balance. So we checked the expression of some canonical targets of this transcription factor, which had antioxidant activities, and we found that there was an increase in the expression of all these genes with dengue virus infection, which indicated that dengue virus leads to enhanced expression of NRF2 target genes. Now, knowing the fact that ROS accumulation is also one of the very crucial characteristic feature of megakaryopoiesis, and taking cues from our transcriptome data, we wanted to see how dengue virus infection could affect the ROS accumulation in the cells, for which, and we found that there was a gradual decrease in the ROS levels in the cells with dengue virus infection in an MOI-dependent manner. And this reduction was comparable to the reduction seen with the treatment of cells with NAC, N-acetylcysteine, which is a very well-known quencher of ROS. We also made use of an inhibitor, dengue virus inhibitor, NITD, and we saw that there was an increase in the ROS levels when the dengue virus replication was inhibited, which made us curious to know that whether the suppression in the ROS is benefiting the virus in some way. So for this, we checked the virus release in the culture supernatant of the same sample set, and we found that there was an increase in the viral replication in cells which showed a reduction in which there was suppression of ROS due to NAC. So this data suggested that dengue virus leads to suppression of ROS accumulation in differentiating cells which promoted viral replication. Now, to further decipher the role of NRF2 in ROS accumulation in the cells and to see the effect of dengue virus replication on it, we made use of two pharmacological modulators of NRF2, and the RA839 and ML385. Now, if you try to understand it like this, during normal treatment of PMA to the cells, there will be an increase in the ROS production in the cells. But as we saw in our data, we saw that with dengue virus infection, this ROS level goes down. If we use an activator of NRF2, RA839, there will be a reduction in ROS production, while if you treat the cells with ML385, the ROS level will go up. Now, given the combined treatment of PMA and RA839, the ROS levels will be less as compared to PMA alone, while there will be a super accumulation of ROS in cells which are treated with both PMA and ML385. Now we wanted to see how dengue virus could actually modulate these conditions and what effect it had on the virus replication. So as expected, we saw an increase and a decrease in the ROS production in cells which were treated with RA839, and the ROS levels were actually reduced by dengue virus infection as well. And simultaneously, we saw an increase in the virus titer in the cells which were treated with RA839 and dengue virus infection. So upon supplementation with ML385, as expected, there was upsurge in the ROS levels in the cells, but the virus was able to reduce this intracellular ROS to some extent. And very surprisingly, we saw that there was a complete inhibition of virus replication in cells in these conditions, indicating that dengue virus increases the NRF2 activity in differentiating K562 cells, leading to a reduction of ROS level and increase in virus replication. Now, to summarize the study, if we see that there is a balance between NRF2 and ROS inside the cell during megakaryopoiesis, but when the cells are infected with dengue, there is disruption of this balance, which causes activation of NRF2, which causes the localization of the NRF2 to its promoter in the nucleus, which causes an activation of the different antioxidant genes, which causes reduction in the ROS inside the cells, which is somehow promoting the virus replication. So, although a very long way to go, given the increasing cases of dengue every year in our country, which causes huge economical burden in developing country like ours, it is very important to have correct therapeutics for the treatment of such diseases. So if we, although a long way to go, if we make use of NRF2 inhibitors to rescue this balance inside the cells, we might be able to just block the replication of virus. So I would like to acknowledge my supervisors, Dr. Milan and Dr. Shankar, for their constant support and guidance. And I would like to thank you all for your kind attention.
This is the work on cell line, K562 cell line, a very famous cell line. Everybody works on that. This NRF story and all, how does it correlate to the human system? I'll tell you the problem in human system. You get dengue infection, and a particular percentage of people go on to develop dengue hemorrhagic fever. And then there is, of course, DHF and shock and this and that die. That could take place. Is there a similar system that how do you correlate your data to the human system? After all, we are concerned with the human health and not the cell line health.
Yes, sir. So actually, there are some limitations of using primary cell cultures where the process of megakaryopoiesis could be studied. And also, as we know that there is a lack of animal models where dengue virus can be studied. So this is a very initial step where we have studied this kind of phenomenon. Definitely, if given time and opportunity, we could just repeat the same data with primary cells and see if this same phenomenon applies.
Is there a similar data available in human system? Because the issue in human is, does NRF lead to leakage of the vascular system? The issue over there is the dengue hemorrhagic fever,
Yes.
DHF, and there is a leakage of the vascular system. Is there a similar NRF2 kind of a system? You're saying you target this and then it is quite okay, right?
So this data that we've shown, it's actually about thrombocytopenia, and we are not studying the last. I mean, the dengue hemorrhagic fever is just like it's the severe condition that we have in patients. Thrombocytopenia, it actually happens in most of the patients affected with dengue. So as far as NRF2 and this the pathway is concerned, so ROS, there is a different kind of there are different theories which are put forward as far as viral infections and ROS accumulation in the cells are concerned. So in some viral infections, the ROS might go up in the cells, as is the case with COVID-19. And the people, instead of using NRF2 inhibitors, are actually using NRF activators. So this is the limitation of this study. And we might not be able to just prove the role of NRF2 in the human system. But there are no reports suggesting that NRF2.
Any reports available, maybe from Africa, where during COVID-19, people also developed dengue infection?
Yes, sir, but that has not been related to ROS or NRF2 as far as I know.
Any more questions from the audience? Thank you, Jaskaran. Moving on to the next, Tarini.
Our next participant is Ms. Khushbu Kaushal.
So good morning to all. I am here presenting the part of my thesis work entitled Associations of Antenatal Maternal Information with Fetal Growth in a Cohort of North Indian Women. So to start with, pregnancy is a 9-month time period, which is broadly defined in a terms of trimester, considering 3 months each. So there are various factors that are responsible for optimal fetal growth. And there are some which alters the fetal growth, which includes the smoking during pregnancy, infections episodes, inflammation, and multiple gestation, which will result in poor fetal growth. So the poor fetal growth is termed as fetal growth restriction. The study in 2012 underwent with 138 low middle income countries indicates that India has the highest rate of fetal growth restriction at birth, which is 36.5%. So the fetal growth restriction fetuses born with a short-term complications of immunodeficiency, prematurity, and organ dysfunction, with a long-term complications of growth retardation, neurological and immune dysfunction, whereas they are at a higher risk of generating metabolic disorders like diabetes. So as it is important to know what are the factors which can be responsible for fetal growth restriction. So one of the important factors that we are interested in looking is maternal inflammation and fetal growth. So the inflammation during pregnancy is the early pregnancy, which is the implantation state, is an increase in inflammation, which reduces during the 2nd to early 3rd trimester, which require the fetal rapid fetal growth. As the pregnancy progresses towards labor, which is again an inflammatory status, which is required for the process of acting delivery, which is parturition. So this is all about the balance of inflammations, which is required for a good pregnancy outcomes. If there is a sudden increase in inflammation due to any reasons, there disrupts the inflammatory balance towards more inflammation during pregnancy, which has been seen in past in poor pregnancy outcomes like preeclampsia and preterm birth. The associations with fetal growth is not still well understood. So, and the data that we have is mostly from a specific time points, majorly cord blood, and the data are lacking from low middle income countries like India. So we hypothesize that an exaggerated inflammatory response is associated with fetal growth restriction. So to study it, we have two objectives: to generate a profile across normal pregnancy and to study the associations of these inflammatory markers with fetal growth. So the current study is inbuilt in an ongoing cohort where pregnant women are enrolled early in pregnancy and followed till delivery, and repeated follow-up visits with repeated biological sample collection, ultrasonography, and clinical data sample collection is carried out from these participants. So first we have seen what are the fetal trends in comparison to the known fetal charts that are clinically used worldwide. So the red one is the fetal growth trends in our population, that is Garbhini, which we can see is comparatively low as compared to all the known charts that are available. We also calculated the proportions of fetal growth restriction in our population and find out that the growth restriction increases with increasing gestational age, which approximately twofold increase in the rates of growth restriction. So next, we were interested in looking at the cytokine profile. For this, we have certain inclusion criteria, that is availability of longitudinal samples and ultrasonography, singleton live term birth with no reported infections and comorbidities. So for this, for estimation of cytokines, we have used a platform Luminex, where we can, with a smaller amount of samples, we can measure a larger number of analytes. So these are 24 analytes that we have measured. The 4 indicated are non-detectable, so were further excluded for analysis. So at different visits, which is the 3 first visit is the maternal time points and the last one is the cord blood. For most of all of the cytokines, we were able to find that there is a significant difference between the cord blood and the maternal time points of all the visits. So to further check it, we have done a principal component analysis, which again shows that the maternal time points, which is all 3 time points, are clusters together, with cord blood having a significant distinct pattern, which another shows that when taking a V1 as reference, we were able to find that most of the cytokine increases at apparently different time points, whereas a cord blood has a distinct profile than a maternal blood. So next, to generate the longitudinal trend across the pregnancy of these individual markers, we have done a linear mixed modeling, where we can, the x-axis is a gestational age and the y-axis shows the concentrations of log concentration of these markers. So among the 20 cytokines that we have chose, the 6 cytokines including IL-18, placental growth factor, VCAM, SFLT1, FLT3 ligand, and IL-4 show a significant trend across the gestation in normal pregnancy. So next, to see how these cytokines are correlated to each other, we have done a heat map analysis and at subsequent visits and find out that there are certain groups of markers that are prevalent across the normal pregnancy at each time point during pregnancy. While irrespective of the visits, there is a strong correlations between the inflammatory markers during every visit. So next, for next objective, where we were interested in looking at associations of the markers with the fetal growth parameters. So to monitor fetal growth, there are different fetal growth parameters like abdominal circumference of a baby, head circumference of a baby, female length, birth weight. So to check this, as we know that, as I shown in this previous slide, that cytokines have a high collinearity, we have taken an approach of identifying a group of cytokines that found associated with the fetal growth parameters. For example, at V1, which is less than 14 weeks, the cytokine marked in red shows the increasing concentration, while the cytokines marked in green has a decreasing concentration. So for taking example like PC5, the group of these cytokines indicated in red as well as in gray combining are associated with a negative head circumference. That means the increase in this cytokine and decrease in this are associated with a decrease in head circumference at birth. Similarly, we have found out different clusters associated with different time points with fetal parameters. So interestingly, we were able to find that there are some of the markers that have a contradictory effect when measured at specific time point. For example, IL-10, which is interferon gamma indisible protein 10, and IL-1 receptor antagonist, when measured at less than 14 weeks, is negatively associated with head circumference at birth. But when they were measured at later time point during pregnancy, which is 26 to 28, as well as in cord blood, they have a positive association. That means they are good for the head circumference at birth. It indicates clearly that the markers have a time-specific associations with fetal growth parameters. So for to summarize what we have done, we have generated a fetal growth trends in a North Indian population, and we found that the Indian population is low when compared to known charts. We have increased rates of growth restriction with increasing gestational age. We have also provided the cytokine profile in a normal healthy pregnancy, and we found that cord blood has a distinct profile with increased inflammatory markers. As well as with this stringent criteria, also we were able to find association of maternal inflammation with fetal growth. But what next? So what next? We are currently doing an analysis where we have two groups of one with having a normal term pregnancy and the one with having a fetal growth restricted pregnancy. We will be doing a cytokine longitudinal analysis to see is there a difference between the cytokine profile between the restricted and a normal pregnancy. And if we are able to find some of the markers that are shown to have a good predictive values, these prediction marker can be used for a proper as a biomarker to early identifications of fetal growth restriction, so that a proper management of this fetal growth restriction can be taken as early as possible. With this, I would like to thank my supervisor, Dr. Shalja Sapuri, and all other members who have helped me in doing this research. Thank you.
बेटा मुझे ये बताओ, ये तो नॉर्मल प्रेगनेंसी में आपने कहा इन्फ्लामेशन अगर मदर में रहेगी तो फीटल ग्रोथ पे अफेक्ट होगा, and that we knew it for many years. Have you been able to take also samples from women who had a history of recurrent spontaneous abortion or family history of recurrent spontaneous abortion? I mean, your hypothesis is certain cytokines drive the growth of the fetal fetus there, and you showed a lot of work, of course, a lot of cytokines that you have studied. And my second question, therefore, is out of the several cytokines that you studied, can you pinpoint 2 or 3 that are of major importance?
So regarding your first question, recurrent pregnancy loss, we are not looking at, because the outcome of interest that we are looking is fetal growth restriction. And in our screening criteria, they are primigravida women because they are first time pregnant. So we haven't included the one with having a history of pregnancy for particular this objective. For a second, I can say there are specific cytokines that are, this one is more important and this is less important, but there are certain like placental growth factors, soluble VEGF receptor, which are actually essential for fetal growth, and they are in a clinical use for a person. For example, I say SFLT and PLGF ratios are in clinical use for early identifications of preeclampsia. And there are many other cytokines that are they have individual roles in optimal fetal growth.
Any questions from the audience? Thank you, Khushbu.
Our next participant is Ms. Pramila Pal.
So good afternoon, everybody. The topic of my presentation is identification and characterization of unknown putative conserved essential transcription regulator in Mycobacterium tuberculosis. So to begin with the broader picture, as we all know that antibiotic resistance is a major problem worldwide, and despite of many clinical practices in use, we are unable to tackle this problem. So when we come to antibiotic resistance in tuberculosis, it's not an unknown phenomena. And as we know that treating this clinically diagnosed form of resistant TB is very tough, especially when we talk about the toxicity of the drugs and the long term of the treatment. there is a surge and need of new drugs and druggable targets in the field of TB, especially after this absurd use of antibiotics in this pandemic era, as we have all seen. So when we talk about Mycobacterium tuberculosis as a pathogen, it is very successful pathogen. And when we look into the peculiarities of this pathogen, we know that it has developed a complex mechanism to regulate a gene differentially when given the different environment, different milieus in the host, and tackle the host arsenals very well. So we decided to study the transcription regulators in the Mycobacterium tuberculosis, which are the topmost hierarchy of the regulatory mechanisms. And out of the 200 transcription regulators known in Mycobacterium tuberculosis, we have filtered out these 20 essential transcription regulators and found out this target RV1830, which is conserved throughout the mycobacterial species and the related genuses, and which is essential in which is essential for mycobacterial survival. And with taking all these things in mind, we hypothesize that this gene RV1830, also known as Erdmann 2020, in the pathogenic strain Erdmann of Mycobacterium tuberculosis, is endowed with a conserved and essential function in the bacterial survival and virulence. So look into its function, we formulated these 4 objectives. And to start with our study, we first wanted to see if this gene is actually important for bacterial survival. And for this, we use CRISPR approach to knock down this gene, since it is essential, so it cannot be knocked out. So via using CRISPR, we knocked it down, and we checked that if this gene is actually suppressed, and by using different ATC concentrations, since the vectors which we have used to overexpress the Cas9 protein and the guide RNA was the Teton system. So at different ATC concentration used, we can see that ATC 50 nanogram per ml was the concentration at which we were getting the good suppression of this gene. And it was checked at the transcript level by QRT-PCR and at the protein level by immunoblotting analysis. Next, we also see that this gene is extremely important for the survival of the bacteria, as shown by this purple line. When we knocked out this gene, the bacteria is not able to grow when compared to all the controls. When we again knocked in this gene, as in complemented the bacteria lacking this gene with episomally expressing this gene again with the tetracycline, with the help of tetracycline, we see that the bacteria was able to survive better, and also the protein levels were restored in the complemented strain. So similar results were also found in THP1 monocyte-derived macrophages, that it is essential for intracellular survival. Next, we moved our study to the in vivo conditions. For this, we infected the mice, 6 to 8 week mice, and let the infection establish for 21 days. After which we divided our mice into 2 groups. One which were given doxycycline and one which were not given doxycycline. So doxycycline here will act as anhydrous tetracycline, which cannot be given to the animals. So after which, at day 42 and day 70, the mice were euthanized and checked for a bacterial load in their lungs and spleen. And as we can see in the result, when the doxycycline was given to the mice which were infected with the knockdown strain, the bacterial load in the lung was very less. When compared to the control, there was no effect of doxycycline. And this is also evident with this lung picture here. We also, not only the bacterial load in the lungs was low, the bacterial load in the spleen was also low when the bacteria was deficient with this gene Erdmann 2020, which shows that there was less dissemination of the infection. So till here, we can conclude that it is important for survival in vitro condition, ex vivo condition, and in vivo condition now. So to look for the mechanism of action of this gene, we did the transcriptomic study. And we found out that there was a global response while we knocked down this gene. approximately 338 genes were getting upregulated and 460 were getting downregulated. So when we segregated them into different functional categories, we found out that the two functional categories were majorly affected and most of these genes were getting downregulated. So as we can see that the one category is of metabolism and respiration. So we did the metabolomic study and correlated it with our transcriptomic data and found out that the pyrimidine metabolism was somehow getting affected, and these genes shown in the red color were getting downregulated, and the substrate of these genes were getting accumulated. We also checked it via phenotype, and we saw that there was a less RNA production in the knockdown cells, and hence the cells were deficient of survival. The next thing which we the next category which was severely affected was the genes which were encoding for the ribosomal structural proteins. And we found, so we checked if there was some lack in the protein synthesis as well. We checked it by puromycin uptake experiment, in which if the less puromycin was taken up by the cells, it means that the protein synthesis will be less in the cells. And we found out that our knockdown strain was deficient in, having less protein synthesis rate than compared to the control. We took SIGA as the non-specific, as a gene, which was the non-specific control for the study, which proves that the less protein synthesis is not due to the essentiality of the gene, but it is the specific effect of the knockdown of RB1830, since SIGA is also essential in Mycobacterium tuberculosis. We check the same thing by doing the ribosomal analysis in these cells, and we found out that there was less of 50S and 70S ribosomes in the knockdown cell when compared to the control. Next, we also did the to find the cognate DNA binding partner of this protein. We followed the SELEX approach, and we found out this DNA is binding to our protein of interest here, and we proved it again by the EMSA experiment, and we screened for this DNA sequence in our transcriptomic data and found out certain genes in which this similar sequence was present in the upstream of the CDS region. Here we can see that some of the genes belonging to the pyrimidine metabolism are having this sequence, and we'll now check if this effect is direct or indirect. To summarize my study, I'll say that if the cell is healthy, all the gene expression is fine, and the bacteria is happy to survive. As soon as we knock down this gene ERDMAN 2020 or RB1830, there is less of RNA in the cell, less of protein, and hence less survival. So it can be a good drug target for future studies. I would like to acknowledge my supervisor, Dr. Nishit Agrawal, REC committee members, all the collaborators, all the lab members, friends, and family. Thank you.
Just tell me this ERDMAN 2020,
Yes.
and there are a number of statements I saw that is very important for survival of the mouse. It affects the protein synthesis, it affects the ribosome synthesis,
Yes.
it affects the assembly in a cell. Is it specific only to the MTB infection, or is it a product that is useful, is used in every other disease or every
So-
other infection?
So as far as this gene is concerned, this gene is only present in the mycobacterial species and few of the genuses like Rhodococcus and other bacteria. So we do not have, as humans, we do not have similar kind of genes, similar kind of gene. So targeting this would not be a problem. And yes, this is specific to MTB since this gene is not present anywhere else.
One of your slides, you use the term MTB is a successful pathogen. Why
Yes,
do
sir.
we call it successful pathogen?
So because the bacteria has developed from the Neolithic age with us, and we have survived with this disease, and it has undergone evolution. It resides in the macrophage, and the cells which are supposed to kill the bacteria, it resides in that cell and obviously tackles it well. It also develops the latency, it undergoes latency, dormancy, and all those things. It has a wide range of nutrients on which it can survive. So it is, so far we know it is the successful pathogen.
So my last question, what is the extent of MDR or XDR TB in India?
Sir, MDR and XDR TB, I'm not very sure of the numbers, but as far as the global burden is concerned, India is one of the, I think, third or fourth number, which has the burden of this XDR and MDR TB.
हाँ, मतलब total numbers में से how many do you think would develop MDR? अगर 100 मरीज हैं
Hmm.
TB के, तो कितनों को have you read somewhere would develop MDR TB or XDR TB? और इसका कारण क्या है?
So I would just guess that the number would be around 25 to 30%, and the reason is the non-compliance. I'm not very sure of the numbers. The reason is the treatment is so long, so obviously there is non-compliance among the patient. So they leave the treatment in between, and again when we start the treatment, the bacteria has already seen the drug and it develops the
No, but
resistance.
I know this is not directly related to your study, but you know, when you work on TB, MTB,
Yes, sir.
you should know each of those things.
Yes,
as
sir.
well,
We
you know.
have,
You have to
I have read it, but
broaden
I'm
your
not
horizon.
recollecting.
And कोई दवाई आई है इसके लिए? Any drug that you know of
So
for MDR?
Sir, Pretomanid is the last drug which was FDA approved for the third line drug, which was FDA approved for treating MDR tuberculosis. Before that, bedaquiline was the drug, and there are many drugs in the pipelines. Although for the first line treatment, we still rely on the drugs which were developed at the
pipeline में तो बहुत हैं. Everybody comes up and CDRI में जाइए आप तो सब कहते हैं कि जी हम नई दवाई लेके आएंगे, लेकिन होती नहीं है. But there is at least one or two that we already know that they are in the use. They're in use.
So
ये
the
थोड़ा सा
most
और भी,
recent
no,
drug is
what I'm saying is
Pretomanid.
try and increase your knowledge
Yes,
base on all of these things, because whatever work you do here has to has a relevance. Translation. आपके
Yes,
तो
sir.
इंस्टीट्यूट का नाम ही translation है,
Yes, sir.
है ना? Okay, thank you.
Thank you, sir. Any questions from the audience? Thank you,
Thank you.
Pramila.
Our next participant is Dr. Ramachandran.
Good afternoon, everyone. Today, I'll be presenting some results from the epidemiological study, which is an observational analytical cohort study evaluating association between antenatal exposure to ambient air pollution and pregnancy outcome. It's a great pleasure to represent an interdisciplinary team from Translation Health Science Technology Institute, Indian Institute of Technology, and Indian Institute of Technology, Madras and Delhi. So, as part of a global effort, all countries in United Nations have come with a set of goals, sustainable development goals, which one of which is that by 2030, we have to end preventable deaths of newborn and children under 5 years of age. And the goals are still a bit daunting for low and middle income countries, with 12 per thousand live births for neonatal mortality and 25 per thousand live births in under 5 age group. If we need to achieve those goals, the interventions should start early during pregnancy or sometimes even before that. And two important approaches are, One is predicting adverse outcomes early so that the current available treatment and preventive strategies can be employed, and identifying modifiable risk factors that can be further intervened upon, either in the domain of maternal nutrition or in the domain of environmental exposure such as air pollution. Air pollution is well known to be associated with chronic illnesses. Air pollution as a risk factor for adverse pregnancy outcome is still emerging. The existing knowledge has shown that there is some association between ambient air pollution exposure and adverse pregnancy outcome like preterm birth and fetal growth restriction. Most of these studies being conducted in low pollution setting, and there are competing risk factors to air pollution such as nutrition and others, which sometimes have not been adjusted appropriately. There are some important unanswered questions in this domain. Is there a safe limit of exposure? Is there a threshold above which complications are remarkably high? Is there a period during pregnancy where women have to be extra cautious? And among those who have been exposed, can we intervene to mitigate the consequences? So to answer these public health questions, we devised two major research questions. What are the critical doses and windows of exposures in the antenatal period to ambient air pollution that increases the risk of preterm birth, fetal growth restriction? And is an increase in exposure to ambient air pollution in the antenatal period associated with some well-known intermediate outcomes such as cervical length and decreased uteroplacental blood flow? So for this study, we enrolled participants who were less than 14 weeks of gestation and willing to deliver at Gurgaon Civil Hospital. These participants are part of a large effort which is called Garbhini Cohort, which is coordinated by THST at Civil Hospital Gurugram, where multiple other questions related to pregnancy are being asked. One of the question is air pollution. Those who are enrolled into this study are followed up periodically to document their outcomes, which is at delivery, birth weight and gestational age at delivery. And during this follow-up, ambient air quality, household air quality data, and clinical and epidemiological data are also collected because all these are interlinked together. In addition, certain other outcomes such as fetal biometry and placental blood flow analysis are done. For exposure to ambient PM 2.5, we all know there are remote sensing satellites, both India and global satellites, which are revolving around the Earth, and these satellites pass through our study area, which is Gurgaon district, once a day. So there are multiple satellites like that. The primary satellite being MERA-2 satellite. So here we collaborated with Professor Sagnik Day in Indian Institute of Technology, Delhi, to collect information from these satellites, which is an air optical density, and convert that into PM 2.5 concentration at a given location. So if we have a given location, geolocation as GPS code, and the duration, we will be able to estimate what is the exposure to ambient air quality for a person who is residing in that area for the given period of time. The outcomes that we were interested were preterm birth, which was defined as any live birth less than 37 weeks of gestation. And cervical length is a well-known pathway towards preterm birth. So we evaluated that also. The second major outcome was fetal growth restriction, which was defined by birth weight and birth weight Z-score at delivery. And placental blood flow is a well-known risk factor. Reduction in placental blood flow is a well-known risk factor for fetal growth restriction and low birth weight at delivery. So these two sets of outcomes were evaluated. So we have 8,000 participants to start with, of whom for an estimated sample size of 3,150, we were able to have more than that sample size for evaluating preterm birth, slightly lesser numbers for fetal growth restriction. So what we are seeing interestingly is we evaluated for different trimesters what is happening between ambient air quality and preterm birth and gestational age at delivery. What we are seeing, as you can see here in the left-hand side, is there is a decrease as we move on. In the x-axis, you are seeing the concentrations of PM 2.5. It starts somewhere around 30, 40 and goes up to 180, 190. So as the exposure increases, there is a decrease in gestational age at delivery. The duration of pregnancy is shortening. And you can see at a threshold of 110, after that threshold, there is a linear decrease with 1 week reduction in gestational age at delivery for every 50 microgram increase in PM 2.5 concentration, which is remarkable. And The categorical outcome, which is, as I said before, preterm birth defined as less than 37 weeks of gestation, the risk starts even at a lower level, but then plateaus at around 80 to 90 and remains consistent after that. That is because we are putting an artificial cutoff at 37, and anything below 37, whether it is even 28, is called as preterm birth. But this clearly shows that there is an association as we move on. Second is looking at birth weight outcomes. Here also, if you can see, there is not, and here, for preterm birth outcome and gestational age at delivery, this association was seen only in first trimester. It was not seen in the second trimester or later during the period of gestation. So there is actually a trimester specific association. When we come to fetal growth restriction, the association is remarkable in the last 3 months before delivery, not in the first trimester or in the mid trimester. And probably this is because the fetal growth mostly happens in the third trimester, and that's where the weight accrues in the fetus. And here you can see at around the same threshold of 110, after which there is a linear decrease in the birth weight at delivery as the concentration of PM2.5 increases. The same is reflected in birth weight Z-score, where it is adjusted for the gestation age. Now, as I said before, these are final events, but there could be something happening mechanistically even before that. In order to interrogate, for preterm birth, we looked at cervical length. Cervical length is a well-known intermediate pathway for preterm birth, and therefore, when we looked at it, again, x-axis has got the PM2.5 concentration, and y-axis has got the cervical length and 30 to 32 weeks of gestation. So there is a linear decrease, saying probably cervical length could be a mediator of the air pollution exposure on preterm birth. Similarly, umbilical artery PI is a marker of placental blood flow. And as the PM2.5 concentration exposure increases, the pulsatility index increases, which means the blood flow is decreasing. Probably this could be a mediator for fetal growth restriction effects that we are seeing. So what do these results mean for pregnant women? Specific periods of vulnerability during pregnancy for adverse outcomes are existing, and any public health policy should be conscious of it. And association between air pollution exposure and certain mediators like cervical length and reduced uteroplacental blood flow seem to be there. Upon proper evaluation, there are drugs currently available to improve the cervical, to reduce the incidence of preterm birth due to reduced cervical length. And there are drugs available to improve the fetal growth if there is uterine artery blood flow, uteroplacental blood flow impairment. So those drugs could be tried. But these things can be done only after the results that we are showing here are validated across different geographical settings, and animal experiments are further done to confirm these biological pathways, subsequent to which randomized control trials should be designed and executed to see these effects. Thank you.
Dr. Ramchandran, there are particular months we know in India when the air pollution is very high. You see it coming up from November onwards, November till maybe February. Now, of course, it is coming down. So, and you said this is an observational study. So there must be several factors that sort of affect the preterm birth or the fetal retardation. You are only looking at one of them, the PM 2.5. The issue is, how did you ensure exposure to ambient 2.5? And what are these participants? Are they belonging to low socioeconomic status or middle or higher? Because the exposure level would vary. Low income wala to bachara sadak pe hai, to usko to, the exposure is much more than a high strata over there and all. Of course, we are taking from the Gurugram Civil Hospital, so there would be more of them as the lowest. But how do you maintain that exposure level, ambient to PM 2.5? That's the critical factor. So that's my point number one. The other that you said was there's an association between cervical length and the uteroplacental blood flow. How did you measure that?
Yes, sir. So for the first question, this cohort is based in Gurgaon Civil Hospital. It's a hospital-based cohort where majority of the participants are from low and middle income population. there is the challenge of confounding is always there, and two major confounders are the socioeconomic status and competing risk factors. For socioeconomic status, we try to adjust by adjusting in the statistical model. We used the participants' education, occupation, and the socioeconomic status as measured by modified Kuppuswamy scale. And the other competing risk factors could be nutrition, and the second one is household air pollution exposure. So household air pollution exposure was asked by whether they use biomass fuel for cooking and whether there is passive smoking exposure during their pregnancy. Those two variables were adjusted. And nutrition was essentially adjusted by BMI at the time of their enrollment into the cohort. Having said all this, a similar as we would expect from any observational study, there will be residual confounding, which would have biased in some way our results. But that's something we need to validate in others.
This kind of a data could create scare in the mind of people unless you do a multivariate analysis, taking into consideration all of those factors.
Yes.
The age of the woman and primary or multi-gravity, or there are a number of factors that would affect the fetal growth, family history, for example. So, for example, इस तरह का डेटा अगर बाहर आ जाए तो केजरीवाल साहब आ जाएंगे सीधे सामने आपके, ये क्या कर रहे हो भाई, ये तो आप बता रहे हैं. हाँ, multivariate analysis इसमें you look into all those factors that affect, and these are known anyway, that affect the fetal outcome.
Yes, sir. Yeah. While we are confident about the internal validity of a study from our data, we are looking at multi-institutional study at multiple geographical locations to validate these findings. Thank you.
Any questions from the audience? Thank you, Dr. Ram.
Our next participant is Ms. Shefali Tyagi.
Good afternoon, everyone. Today I'm going to give my presentation on the topic host immune correlates of disease persistence in TB. So to start with the introduction, Mycobacterium tuberculosis, the causative agent of TB, was discovered by Robert Koch in the year 1882. And even after 2 decades of discovery of this bacteria, BCG vaccine came into being, followed by the anti-TB drugs in another 2 decades. So in the current scenario, the major threat is posed by the emergence of drug-resistant TB, that is the MDR and XDR TB. And even with the advancement in science and technology, after a century, we are unable to reach our goal of eradicating TB. So TB is an infectious disease, and based on the pathogen, we can classify the infectious disease into 2 different categories, that is acute infection and chronic infection, based on 2 very different outcomes. So traditionally, we have acute infections with fatal outcomes such as plague and anthrax, which are highly virulent and have high mortality rate. And I'll classify such pathogen as not so smart pathogen. On the other hand, we have chronic infections such as TB, which have traded off its virulence with its transmissibility, and it is therefore one of the infectious disease in the world. And the major characteristic of TB infection being its adaptability to host environment, leading to long-term disease persistence. So talking about the bacterial persistence, first I'll talk about the antibiotic persistence, which is the bacterial population that is refractory to the antibiotic treatment, whether in vitro or in the host. And then we have the disease persistence or the persistent infection, which is not efficiently cleared by the host. And it involves the immune evasion strategies by the bacteria. So both these virtues contribute to a slow growth rate and metabolically inactive population, which is drug tolerant and are called as the persister cells. So in this talk, I'll be focusing on the persistent infection or the disease persistence. So during a conventional TB infection in a mice model, the TB grows exponentially during the first 3 weeks or 4 weeks post-infection, and then it goes into a sleep mode with a live and a let live let live policy. So we were interested in studying any mutant which does not follow this common trajectory. And to our surprise, we had a mutant in our lab which survived better than this wild type, as can be seen with the bacterial load is highly significant in this mutant strain in the lungs 2 weeks, 4 weeks, and 8 weeks post-infection, as well as in the spleen 4 and 8 weeks post-infection. Also, when we did a gross histopathology of this mutant, we found that the granulomas was significantly more in this mutant strain as compared to the wild type, and the granuloma score revealed the same with significantly high granuloma score in this mutant as compared to the wild type. When we did a high MOI of infection, we interestingly found that in the case of the mutant strain, almost 80% of the mice succumbed to TB disease. However, in the case of the wild type, all the mice survived till 16 weeks post-infection. So we were interested in knowing that what are the immune cells or the immune marker that is contributing to such drastic phenotype. So to study the same, we did the immunophenotyping in the mice model of TB infection by infected the mice with the wild type and the mutant strain, and we did immunophenotyping at 2 weeks and 4 weeks post-infection. So firstly, we looked at the innate immune markers. So monocyte, which are the leukocyte that circulates in the blood and the spleen, was significantly less in the mutant strain, as can be seen by the green bar as compared to the wild type and the complemented strain. However, monocyte, which get differentiated into the macrophages, which is the primary site of TB infection, were significantly high in the mutant strain, both 2 weeks and 4 weeks post-infection. Also, the MDSCs, which are the myeloid-derived suppressor cell and are the innate suppressor cells, which act by inhibiting the T cell response, was significantly high in the mutant strain, both 2 weeks and 4 weeks post-infection. The infiltration of neutrophils in the lung is a common feature in severe TB patient as well as in the susceptible mice. And also in our mutant strain, we found out that these cells were significantly high 2 weeks and 4 weeks post-infection. Natural killer T cells, which forms a bridge between an innate and adaptive immune response, were also significantly high. However, we didn't see any differences in the natural killer cells. So we also further looked at macrophage polarization. So macrophages can be classified as M1 and M2 depending upon the stimulus. So upon interferon gamma or LPS stimulus, macrophages are called to be classically activated or M1 macrophages, and upon IL-4 or IL-13 stimulus, macrophages are called to be M2 specific or alternatively activated. So the M1 have pro-inflammatory function and are microbicidal in action, and these are the markers of M1. And M2 are anti-inflammatory in their function and are immunosuppressive, and these are the markers of M2. So we did qPCR analysis to check the M1 and the M2 markers, and although we found that the fold change expression of the M1 markers such as NOS2 was significantly high in the mutant strain, however, all the M2 markers like ARG1, FIS1, and YM1 was significantly high in the case of the mutant strain. Also, we looked at the TH1 and the TH2 markers, and we found out that IL-10 and IL-6 TH2 markers were significantly high in the mutant strain as compared to the wild type and the uninfected. Further, we looked at the acquired immune response marker. So firstly, we looked at the CD4 helper T cells, and we found that they were significantly downregulated in the mutant strain in the lungs, spleen, as well as in the lymph node. And also the CD8 cytotoxic T cells were significantly down in the mutant strain in the lungs, as well as in the lymph nodes. So further, we wanted to see that what pathways may be involved in such phenotype that might be contributing to it. So to have a deeper insight into this, we did a host transcriptomic profiling by isolating the RNA from the lungs of the infected mice 4 weeks post-infection. So this is a heat map of the same, showing the genes that were upregulated in the mutant strain by the red, and the genes that were differentially downregulated in the mutant strain as compared to the wild type with the green. And this is the volcano plot for the same, showing the genes that were upregulated and downregulated. So when we did a pathway enrichment study, we found that mostly genes that were belonging to the inflammatory pathway as well as neutrophil-associated pathways were significantly upregulated in the mutant strain. So to summarize, a macrophage which is the primary site of TB infection. When the MTB is inside the macrophage, it releases certain immunomodulatory or immunostimulatory signals. So in the case of our wild type, which grows at an exponential rate in the initial stages of the infection and then goes into the sleep mode, we found out that there is a controlled bacterial growth resulting in a discrete granuloma and had immunostimulatory signals, which is leading to long-term disease persistence. However, in the case of our mutant strain, there were more Th2 signals with high bacterial load, diffuse granuloma, and they were immunosuppressive, and then hence it failed to persist. So these were the immune cell populations that were significantly high in the mutant strain, that is the neutrophil, the M2, and the MDSCs. And thus, studying this differences or the immune cell that are contributing to this phenotype will help us in understanding the mechanism of disease persistence and also in designing the novel strategies for developing host-directed therapies. So I'd like to acknowledge my supervisor for his constant guidance and support during the study, all my DAC members and all the collaborators, my lab members and technical staff, THSTI, and my funding agency. Thank you all.
Shefali ji, this cytokine skewing to Th1 or Th2 has been known in the
Yes,
past
sir.
from many years, so many studies. What additional did you find through your transcriptomic studies?
Okay, sir. So, sir, this is, as you correctly pointed out, this is already known in the phenotype.
You have to convince me that you are not doing omics for the sake of doing omics,
Okay.
but you did it because you wanted to find something new.
So, sir, what is the novelty of our study? First, I like to point out that it is already known such phenotype like neutrophil infiltration, MDSC, high rate is known to be there in the TB patient. However, we have a mutant that is showing all these characteristics. So, like this single mutant is contributing to all these phenotype is a novelty of our study. And secondly, knowing these pathways, if we will further get into the metabolomics or if we get a particular metabolite and we can just reverse the phenotype of this mutant strain. So this could be used to target the persister population. And so that's the novelty that I would like to point out.
What translational value?
Sir, so translationally, this is like a long way to go, but yeah, as I said, to be honest, but as I said, like we will also like to
I
do the-
like your honesty.
Thank you, sir. So I would like to say that we will also do the metabolomics of from the serum of these mutant and the wild type infected mice, and then we will look whether we could get any particular marker that could be used to reverse this phenotype, and then hence there's a very long pathway to be taken it into translationally.
Any questions from the audience?
Host-directed therapy,
Yes, sir.
but the effect which you are seeing is in the background of toxin-antitoxin mutant. So to generalize this as a host-directed therapy, how would you rule out the toxin-antitoxin effect?
So, sir, like we will do, and like if we'll study any metabolite or anything, we'll do it in both the wild type as well as the mutant strain, and then we'll take it further. So I didn't
But
understand your
the
question.
effect, what you are seeing of the hypervirulence
Yes.
could be because of the knockout of the toxin-antitoxin, right?
Of this gene, right?
Of this gene, which
Yes,
is
sir.
probably antitoxin, I believe.
Yes, sir.
So if you want to generalize it as a host-directed therapy, I'm not able to understand how this study can lead to that in that direction. Because this is very specific to toxin-antitoxin.
Yes,
Either you
sir.
can target the toxin-antitoxin.
This can be also used, but
How
like
the host-directed therapy can
from
be,
my from my study, what our approach would be that like we are getting the neutrophil in the study, we're getting the neutrophil.
So in another word, if I put it like, say, if you might be having some other mutants showing hypervirulence.
Yes, sir, there could be
Can we
redundancy.
expect the same profile of the host cytokines in those also?
So that is quite possible that there must be redundancy, but any such
doubt on
phenotype,
that.
yeah, like you're right that it could be possible that there is redundancy in this phenotype, but any such phenotype in the case of any immune profiling is not reported with such phenotype in any mutant strain till now in TB. So, yeah.
There are a couple of mutants which are hypervirulent, which are reported.
But like immunoprofiling with
Yeah, that has not been done.
that has not been done. Yes, sir. Yes, ma'am.
in this
So
particular?
ma'am, we have already like published an article regarding the function of this mutant, which is published in M System. So this is a RNase toxin, this mutant, which targets a pro-tRNA, and that has been mechanistically shown already. And I have carried forwarded this work to see why there is this hypervirulent phenotype in this case of the mutant.
So is it just one protein which is mutated in the mutant? Or
Yeah, we have just taken,
one?
yeah.
Okay, can that be, can that act as a target for small molecule inhibitors?
Yes, yes, ma'am, sure. We can study it as a target for targeting the persistent population.
Thanks.
Any more questions? Thank you, Shefali.
Thank you so much.
And our next and final participant is Ms. Tanuko Sen.
A very good afternoon to all of you. I am Tanu Priya, working with Professor Ramandeep on tuberculosis, particularly analysis and characterization of type 4 toxin antitoxin systems from Mycobacterium tuberculosis. So to start with the brief intro, tuberculosis is an aerosol infection which is caused by the bacterium Mycobacterium tuberculosis that resides inside the alveolar macrophages. And upon infection, the host develops characteristic lesions called granulomas in order to contain the bacteria. So inside the host, the bacteria is exposed to various stressful. Sorry. So inside the host, the bacteria is exposed to various stressful conditions like low pH, lysozyme, reactive oxygen species, nutritional starvation, hypoxia, as well as antibiotics. Yet the bacteria is able to survive successfully inside the host. And one of the major factors that help the bacteria to survive inside the host are toxin antitoxin systems. So these systems are basically bicistronic operons in which the antitoxin lies upstream of the toxin gene, and in normal conditions, the toxin activity is being taken care by the antitoxin. However, under stressful conditions, the antitoxins are degraded that releases free toxin, and this targets essential cellular processes, thereby shutting down the bacterial metabolism. So based on nature of these these molecules, whether these are RNA or proteins and their mode of interaction, they have been classified into 8 different types, and I'll be focusing on type 4 toxin antitoxins, where both these molecules are proteinaceous in nature and they bind to a common target. So it has been shown that these systems in E. coli, they target the cytoskeleton machinery and thus inhibit bacterial replication, and they also protect bacteria from phage infection. Recently, it has been shown in mycobacteria that the antitoxin neutralizes the toxin activity by phosphorylation mechanism, and the toxin targets the bacterial translational machinery by inhibiting tRNA acceptor systems. Yet their role in stress adaptation, physiology, and disease pathogenesis is not known. So in MTB, there are a huge number of TS systems, and among them, the type 4 family was recently identified. And in these systems, the toxins belong to the nucleotidyl beta polymerase families, and they specifically bind GTP. So in these systems, the toxin have 4 conserved motifs, whereas the antitoxin has a functional N-terminus domain, which is involved in transcriptional regulation, whereas the C-terminus domain is involved in toxin neutralization. So in MTB, there are 4 annotated systems, and I'll be focusing on these 3 systems, when A, T2, T3, and T4, where A refers to the antitoxin and T refers to the toxin. So accordingly, my work was divided into two major objectives. One was to functionally characterize these systems, and other was to investigate their role towards MTB physiology and virulence. So I started off with the sequence alignment of these proteins from MTB with other homologs present in bacteria. I found 4 conserved motifs, as I stated before, and also the phylogenetic analysis showed that these are closely related to Acidifilum or Helicobacter. So when we overexpress these proteins in E. coli, we found that MT2 was not toxic. However, MT3 and MT4 inhibited bacterial growth in a bacteriostatic manner. And the growth was restored upon co-expression of only the cognate antitoxins, and no crosstalk was observed within the system. So this was also seen through our spotting assays, where we can see that only MT3 and MT4 inhibited bacterial growth, which was restored by the co-expression of the cognate antitoxins. So next we came to mycobacteria, and using ATC inducible system, wherein the addition of ATC relieves the repressor from the operator, and it induces the gene of interest, which is cloned downstream. So in accordance with our E. coli results, we found that MT3 and MT4 were active as compared to MT2, and our CFU assays also revealed that they inhibited the growth in a bacteriostatic manner. So as I said, that these proteins are involved in inhibiting the cytoskeleton machinery, and we tagged these proteins with GFP and studied their localization patterns using Mycobacterium smegmatis as our model. So as compared to EGFP alone, we found no localization pattern in MT2, which was an inactive toxin, whereas in MT3 and MT4, we found either mid-cell or polar localization of these proteins, indicating their interaction with either cell division proteins or cell elongation proteins, which are present at the poles. So next, to study the role of these active toxins in MTB physiology and disease pathogenesis, we simultaneously deleted these two toxins from Mycobacterium genome using recombinant homologous recombination, which was verified with PCR. So I studied the growth pattern of this double mutant strain. It did not alter, the deletion of these proteins did not alter the growth pattern of MTB, also the biofilm formation, also the colony morphology. However, when we studied the effect of deletion on the virulence of mycobacteria in mice, we found that this double mutant strain was severely attenuated at 4 weeks and 8 weeks post-infection, both in lungs and spleens. Similar results were observed in guinea pig as animal model, and our results were also validated through the gross pathology data, where we can see that the lung anatomy was restored upon deletion of these proteins at 4 weeks and 8 weeks post-infection. So because we saw this interesting data that deletion of these two genes could attenuate mycobacterial growth, so we tested this strain as a vaccine model in mice as well as in guinea pigs. Interestingly, we found that in mice, the double mutant strain acted as good as BCG at 4 weeks and 8 weeks post-infection. And moreover, in guinea pigs, we found that this strain acted better than BCG at 4 weeks and 8 weeks post-infection, and which was also seen through our gross pathology data. So in summary, I would like to summarize this work stating that overexpression of MT2 did not inhibit the bacterial growth, whereas MT3 and MT4, they inhibited the growth in a bacteriostatic manner. And MT3 and MT4 showed mid-cell or polar localization in M. smegmatis, and deletion of MT3 and T4 severely attenuated bacterial survival in animal host tissues, and Typho-DM acted as potential drug target and prophylactic vaccine candidate against tuberculosis. So I would like to acknowledge my group, Professor Ramandeep, who is my supervisor, Dr. Krishan Gopal, our collaborator at IMTech, my DC members, my lab members, and lab attendants and BSL3 staff at UDSC and ICGP and DBT for funding. Thank you so much.
Tanu.
Yes, sir.
Tell me how much this work on MEN3 and 4 has already been done in literature?
In mycobacteria, sir, only...
You showed us the work on NPM, like the M mathematics.
my work was on smegmatis and MTB plus E. coli. So initial studies came up from E. coli only, where they only showed that these proteins inhibit bacterial replication by targeting cell division proteins. Then in mycobacteria, the first, like the very initial reports came up with only the structural studies where people have solved the structure of these proteins. And then recent studies that I showed, they have stated that one of those proteins, they were toxic, and we also got similar results, but they showed that they discussed the mechanism, wherein they showed that the antitoxin neutralizes the toxin activity through phosphorylation of important residues present in the toxin. And the other study showed that this toxin actually targets the tRNA present in MTB. So none of those studies showed any role in the virulence of, like a role of these proteins in virulence. And actually we found some interesting data that the mycobacterial strains were attenuated. So I have shown only double mutant work here, but I have also done like single mutants, and all my mutants are not able to cause that much disease in host tissues. In
Because
humans?
humans?
No, sir.
No, it's just the, the
In
cell lines that you
prokaryotes.
showed. So how do you take it forwards, this
Sir, actually,
study?
I want to study this, like, I want to study whether study these systems at relative clinical markers in the clinical patient samples, and also take, because this is just an initial very preliminary data. So to develop is a vaccine model, we might have to unmark the antibiotic resistance cassette that we have used to replace the ORF. And further, we can also study various regimes, like in combination with BCG or any drug chemotherapy, which is also available. So various combinations can help us to, take this forward in future plans.
Thank you, sir. Any questions from the audience?
So Tanu, very excellent talk. So what is your take on multiple TA systems? You said like there are 70 or so TA systems in mycobacteria.
Yes.
So what do you think about them? Like how one can, convince that what is the utility of so many TA systems, and all of them are seemingly important?
Okay. So the very first thing which I like, when I went through the literature, I saw that Mycobacterium smegmatis, which is non-pathogenic and fast growing, it has only 3 systems, whereas this MTB has 70 systems, around 70, 80. So there might be some hidden mechanisms where these systems are quite stress responsive. So we have also studied when we
if
have.
I talk about the recent one where the tRNA, was found to be regulated by one of these. Do you think that this is a kind of conserved function across other bacteria also?
This kind of?
Is a conserved function of this particular toxin, antitoxin, in other bacteria than mycobacteria?
Actually, this conserved, it could be, because in type 2 systems, it has been shown that in.
I think you must have done the phylogeny and the conservation analysis across outside mycobacteria.
No. The function for type 4 has, as tRNA targeting, this has only been studied only in E. coli and MTB, not across the across other bacteria.
So do they conserve, like the function is conserved?
It could be, yes.
No,
Because of because it has those conserved motifs. So this function might be conserved, but studies have been done only in E. coli and MTB, not across. For tRNA target, like that, as the tRNA targeting studies have been done only in E. coli and MTB.
Okay. I think you must be knowing that what is the reason for bacteria having so many TA systems. So
okay. So when these systems they target, so it has been shown that they particularly target translation systems. So it might help bacteria to shut down its metabolism by inhibiting important proteins that could be otherwise target for, like, for example, antibiotics, or which could be bactericidal in nature. So shutting down those important metabolic pathways might help the bacteria to escape from the host immune response, or the antibiotics, which could be antibacterial.
Thank you, Tanu. Any more questions from the audience? Thank you, Tanu.
Thank you.
Thank you everyone for your lovely presentations. We are also joined here today by college students from different colleges across Delhi and Faridabad. And now it is time to commence with their quiz. So to start with that, I would request all the quiz organizers to step forward to the stage. And I would also request 4 members from each team to step forward to the stage as I call out your name. So team A will be Maitri College. You can just queue right here. Team B will be Manav Rachna International Institute of Research and Studies, and Team C will be Venkateshwara College.
Hello. And I want to ensure that according to everybody. What is this going on? Okay. When did he tell you the morning? Not before that. And you are that I gave him an order to do this.
Hello. टेबल मैं रोटेट कर देंगे बस अगर 2 हो जाए सो माइक ए से जाओ
तो जरूरत नहीं
नहीं.
ऑन है राइट दोनों ऑन
So now it's time to start the quiz competition. We have 3 teams with us. Team A from Maitri College, Team B from Manav Rachna College, and Team C from Venkateshwara College. So give them a huge round of applause. So we have 2 rounds in this competition, and we'll be starting with the first round. And we have given you the instructions on the sheets. I hope you have read the instructions, and I'll be repeating the instruction for first round. So in round 1, the questions will be based on general science, and there are 2 multiple choice questions and 2 picture-based questions, which we will be asking from each team. That means we have 4 questions for each team. And 1 question will carry 10 points, and the time limit for answering each question is 45 seconds. First, we will be reading the question, and then after that, we will be starting the time for you to answer. And for answering 10 points, and for incorrect answer, 0 points. Now we'll begin the round 1. So first question for team A, that is Maitri College. Which of the following techniques cannot be used to visualize DNA? A, gel electrophoresis, B, next-gen sequencing, C, electron microscopy, D, X-ray crystallography. Your time starts now. Team A.
It's either C or D. I think it's X-ray.
Be careful before answering.
20 seconds left.
20 seconds left.
15 seconds remaining.
So your answer is team A? C. Sorry, it's incorrect. So
Audience,
audience would like to guess?
Akshay.
Absolutely
NGS.
correct.
Okay.
Okay. Now next question for team B, Manav Rachna College. Which of the following branches of biology is concerned with the effects of radiation on living organisms? A, actinobiology, B, cryobiology, C, radiobiology, D, aerobiology. Your time starts now. No need to say.
It's radiology, C.
Sorry, incorrect answer. Audience would like to guess?
Anybody from the audience?
Anybody?
Sorry?
No, it's incorrect.
Yeah.
Correct answer. Okay.
2 answers are wrong.
Okay, now next question for team C, Venkateshwara College. What is the medical name of black fungus? Kala azar, mycorrhiza, mucormycosis, mycormucosis.
Mucormycosis. C. It's C.
C. Yes, it's the correct answer. 10 points for Venkateshwara College. Now we'll start again from team A. A person exerts the most pressure on earth at A, sitting position, B, standing position, C, lying position, D, chakrasana. So your time starts now.
Standing position.
Yes, it's the correct answer. 10 points for team A. Now moving on to team B. What is the second closest star to Earth? A, Proxima Centauri, B, Alpha Centauri, C, Polestar, D, Sirius. Your time starts now. 20 seconds left.
A Proxima.
Yes, it's the correct answer. 10 points for team B. Now, next question for team C. The symbol for tin is SN. What does it stand for? A, sternum, B, sternum, A, sternum, D, stitinum.
Option A, sternum. Option
Sorry, it's incorrect. Anybody in the audience would like to guess? Yes, it's correct. Okay. Now we'll be starting the next series of questions, which are picture-based questions, and the rules are a bit different. Each question will carry 10 points, and the time limit is still the same. And we'll be showing you a picture. It is of a famous scientist, or it can be of a phenomenon. You have to guess the phenomenon or the scientist. Now, the first question for team A is, name the Indian scientist who was awarded the Nobel Prize for Physiology or Medicine in the year 1968 for the interpretation of the genetic code and its function in protein synthesis. Your time starts now. You can use the hint or identify the picture over there, the statement written. Fifteen seconds left. Time's up, team A. Anybody from the audience would like to guess? Yes.
One moment. Shall we pass?
Okay, sorry. It will be passed on to the next team. And team B, what is your answer?
It's Hargobind Khurana.
It's correct. Extra 5 points for team B. Now, the question for team B. A linen draper turned microscopist whose discoveries helped lay the foundation for sciences of bacteriology and protozoology.
Anton von Leeuwenhoek.
Yes, it's the correct answer. 10 points for team B. Now, the question for team C. This Indian scientist is known as the father of radio and wireless communications. Team C, your time starts now.
Raman, Dr. Raman.
Sorry, it's incorrect. Team A, your answer, it's the chance to get extra 5 points. He's a famous Indian scientist. Shall I pass it to the audience? Do you know? Okay. Anybody in the? Yes, a correct answer. He is J.C. Bose. He has a famous institute in Kolkata named after him. Okay. Now the question for team A. Name the duo of exceptional leaders and scientists who were the father of Indian space research program and nuclear science program. Team A, your time starts now. Indian Space Research Organization, ISRO. He was the founder of ISRO. Ten seconds left. Team A. Sorry, team A. Time's up. Team B, would you like to guess?
Vikram Dharabhai.
Repeat the answer.
Vikram Dharabhai.
You are not correcting the surname.
Vikram Dharabhai.
And the other personality? The other personality in the picture? You named only one. Should I pass on to the audience? No, no. It will be passed on to only one team. So, okay, anybody in the audience would like to guess? Yeah. Absolutely correct. Now, the question for Team C. Name the Indian Nobel laureate who found that when light traverses a transparent material, some of the deflected light changes in wavelength. Your time starts now, Team C.
It's C.V. Raman effect.
Absolutely correct. 10 points for Team C. Now it's the end of round 1. We'll be starting with the round 2. Scores. And the scores after round 1 are Team A on 10 points, Team B on 25 points, Team C on 20 points. So Team B is leading, followed by Team C and Team A.
Now we are proceeding towards the round 2, and it will be a buzzer round. The question will be presented on the screen, and the team who presses the buzzer first will answer. So be quick. The time limit for answering is 15 seconds in this round. For every correct answer, you will be rewarded with 10 points. But if you are going to give the wrong answer, the -5 points will be deducted from your previous round 1 scores. The team with the highest scores will be the winner of this quiz competition. So now we are presenting the question. The new falsely identified COVID strain named Delta Cron was identified originally in which country? Now the time starts now. No one? Audience. Now we are passing this question to audience. Is there anyone who would like to answer? No, 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 no. Right. Right. Now, next question. CAR T cell therapy, which is often seen in the news, is used to treat which disease? Anyone? Okay, we are again passing it to the audience. Anyone? Okay. Correct answer. Now we are going. Now the next question. SpaceX announced a new partnership with which technology company to strengthen use of its satellite internet connectivity? No. Now, -5 for the team A. Who was the founder of the Serum Institute of India?
Now again, we are passing it to the audience. Anyone? Okay, correct. The next question on the screen. What animal was first cloned using the technique somatic cell nuclear transfer? Yes? Okay, correct. Now plus 10 points for the team C. The next question on the screen. Which institute has set up the Artificial Intelligence and Robotics Technologies Park in India? It's a popular institute. You might have heard of this institute. Passing it to the audience. Correct answer. Again, the next question on the screen is, what is the name of the NASA program planning to land humans on the moon by 2024? Any team would like to answer? Now again, passing it to the audience. Anyone? Right, correct. The next question on the screen. The last question, you have still chance to score plus 10 points. What is the name of the first indigenous kit approved by ICMR for the detection of Omicron? Any guesses? Again, passing it to audience. Correct answer. Now we are wrapping this round and we will be telling you the scores, the final scores. So the team who has won this competition, first prize, the team C. Clap for the team C. And they are followed by the... And now it is followed by team B with 25 marks and Manav Rachna. And last we have the third winner and that is team A with plus 5 marks. Maitri College. Thank you.
For this, I would request our chief guest, Professor N.K. Mehra, Director of THSTI, Professor Pramod Garg, and Dr. Shinjini Bhatnagar to kindly step forward for the prize distribution. We'll be announcing the prizes for the oral presentation first.
The first prize goes to Tanu Gosai. Can we have Tanu here?
The second position is shared by Ms. Jaskaran Kaur and Dr. Ramachandran. Ms. Jaskaran?
Anybody from Jaskaran's lab?
Anybody that could accept? And for the quiz, our winners are from Venkateshwara College. Second is
Tell 8 and 6, no? Tell 8 and 6, no?
The winners from Venkateshwara College are Bhavna, Rhythm, Palak, and Vedahi. Can we have a huge round of applause for the team? The second position goes to Manav Rachna International Institute of Research and Studies, to Samradrata, Savan, Tanvi, and Priti Bhavanvik. Let's keep clapping for them, guys, please. Okay. And the third position goes to Maitri College. It goes to Madhur, Radhika, Pooja, and Neha. Yeah. Huh. Third position goes to Maitri College. Yeah. And we have Ms. Jaskaran with us now. She has won the second position in oral presentation. Thank you, sir. Now, I would like to request Dr. Tarun Sharma to come forward for the vote of thanks. And I would also urge the college students to stay here for further instructions. Thank you.
Honorable Chief Guest, Dr. N.K. Mehra, respected Executive Director, THSTI, Dr. Pramod Garg, Professor of Eminence, Dr. Sanjini Bhatnagar, THSTI Society member and DBD officials who have joined online, faculty and students from Hansraj, Maitri, Sri Venkateshwara College and Manav Ratna University, dear fellow scientists, faculties, research fellow, students, and everyone present here in person or virtually, a very good afternoon. It is my proud privilege to propose a vote of thanks and acknowledge the contribution of all who worked relentlessly to make this event possible. I, Tarun Sharma, on behalf of THSTI and entire fraternity of THSTI, extend my sincere thanks to Almighty to make today's event a grand success. On behalf of THSTI, I extend my hearty vote of thanks to our chief guest, and Dr. Mehra, who spared time from his busy schedule to grace this occasion. Sir, the keynote address that you made, it was very informative and it has really enlightened us. And the journey that you have shared, that was very remarkable. And I would like to thank you for that. And an event like this cannot happen overnight. The wheel started rolling a week ago. It required direction, mentoring, and hard work. We are fortunate to have great leaders like Dr. Garg and Dr. Sanjini, who are being the catalyst that motivated us to do our best. And sir, I also thank you for nicely highlighting the achievement of THSTI and capability of THSTI during your address. I also want to extend thanks to PhD scholars who have presented their respective research work. The quality of work they have presented clearly reflected the efforts and dedication of these scholars and their mentors also. I must mention our deep sense of appreciation to Dr. Amit, Dr. Pallavi, and Dr. Amit for conducting such an engaging science quiz. And at last, I would like to thank everyone, THSTI administration, IT team, and everyone who has directly or indirectly contributed towards the success of this event. And last but not the least, a big thank you to each and everyone gathered here and graced this occasion with their presence. Thank you very much and Jai Hind. And a small announcement that now you can proceed for the lunch, and after lunch, kindly assemble in the dome area, and then respective faculty will take you for the lab tour. And now please stand up for the national anthem.
भारत भाग्य विधाता, पंजाब सिंध गुजरात मराठा द्राविड उत्कल बंगा, विंध्य हिमाचल यमुना गंगा उच्चल जलधि तरंगा, तव शुभ नामे जागे, तव शुभ आशीष मांगे, गाहे तव जय गाथा, जन गण मंगल दायक जय हे भारत भाग्य विधाता, जय हे जय हे जय हे जय 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 हे।